Hello, everybody. Welcome to the stream. Welcome. I am Phi the Neutrophil, your white blood cell VTuber extraordinaire, here with another stream. Hello. Let's start off by just saying hello to everybody. Hello, Hero. Hello, PogChamp. Hello, Hungry. Hello, Will Not. Hello, Amaryllis. Hello, Marlon. Welcome. Hey, Cleo. Welcome. Hello, Guy. And who else do we have here? James. Welcome. Seed. Welcome. Hello. Today, we are going to be talking about the molecular biology of cats. Very exciting, a stream that I've been planning to do essentially since January, when we met the incentives for the glioblastoma stream. So the entire reason why this is happening is because of tremendous support from this here community. So I want to thank you all to begin with. If you're unfamiliar with what I'm talking about, let me just remind you that in January I had a glioblastoma stream, so a stream that was dedicated entirely to the science and treatment of this deadly form of brain cancer called glioblastoma. This was a 12-hour charity stream, a marathon stream, in which a lot of people from this community donated, and we managed to raise up to $1,000 for St. Jude's research. And that has been an incredibly meaningful experience for me, which is why I'm also doing another 12-hour charity stream in January. And one of those incentives was to do a stream where I would talk about the molecular biology of cats. So thank you to everybody who helped make this happen. I'm super excited to get into this. Yeah, so this stream would not happen without your generous contributions to St. Jude. Oh, hey, surprise fans, welcome. I'm gearing up for another monster 12-hour charity stream in January, this time for lack of a better term, female reproductive health. I'll be covering ovarian cancer, breast cancer, and oh my gosh, I always forget what this last one is called. Endometriosis. took me a while. Took a while for the gears to turn. I love those emojis. That was so cute. So what do cats have to do with brain cancer? The charity stream that I did about brain cancer, one of the incentives, I don't remember what the number was, but I was like, if we raised $200, I would do a stream where I covered the molecular biology of cats. Okay. So there's another one of these happening in January. I'm still planning it out, but I'm very excited to do another one of these monster streams because I had a lot of fun last time. If you have any stream incentives, like milestones for us to hit, what I could do to help energize the community to help out with another one of these very important clauses, let me know in chat or in the Discord, which you can get access to by typing exclamation point Discord. All right, with that preamble part 1 done, we have a preamble part 2. So this is a stream about the very valuable contributions that cats have made towards our understanding of neuroscience. And I don't want to dance around this topic. This involves animal experimentation, right? This is not without cost, although you might find it a little happier to know that this kind of research done in cats is not really as popular as it was back then. We now have better tools that we can use to sort of patch up the weaknesses of other animal models. But I wanted to kind of spend a slide just acknowledging that biology isn't vegan, right? That the cost of human progress often comes at a loss to, some other entity. In this case, it is research animals. But I do want to make a note here that it is important for me and all of us to acknowledge that laboratory animals do not have the best lives, right? And it's important to remember this because if you are planning to use a research animal, it should be for a very good reason. I've repeatedly reminded people that there are strict standards, at least in the US, to limit the number of animals that are needed for research, right? So before you go off to do your experiment, you have to fill out these forms that are hundreds and hundreds of pages long, basically making the argument that yes, I absolutely need to use a research animal, and I am very aware of how many I will need, so you don't just waste animal lives for no good reason. And again, something optimistic to look forward to is that more powerful technology has reduced the need for cats as a model organism. Now, back then, when this research was done, the number of molecular tools that were available were not as varied or powerful. And so, today is about remembering the major contributions that cats have made here, right? Thank you, cats. Yeah, exactly. This is important so that we don't take these animal lives for granted. So thanks to the contribution of these cats, we have accelerated our understanding of the development of the visual system, visual processing, and for me, what's most striking is cerebral plasticity as a whole. So not only did we get a bunch of information about the eyes and the way that the eyes are connected to the brain, we also learned a whole lot about just how the brain functions, which is really, really cool. And with that double preamble out of the way, let's actually get into it. So in the 1950s, two guys started a 20-year partnership to study the visual system. Oh no, why are my images not loading? Oh, give me 1 second. That's so weird. Give me give me a second. Let's hide this. Wow. I don't remember having this bug before. So let's just reopen the slide and hopefully everything will be fine. Gosh, that's okay. Are we chill? Okay, cool. So let's do that again. Not all the images too, like the title slide was fine. Okay, let's rewind and get this image back up on the screen. Okay, there we go. Weird PowerPoint. Very cool. Okay, yeah. So two guys started a 20-year partnership to study the visual system. These two people were David Hubel on the left over here and Tornstein Wiesel on the right. I think what's cool is that Wiesel is actually still alive today, which is nuts. He's 101 years old, which is so crazy. So you can imagine just like the state of molecular biology from when he was born to the current year. It's incredible. He was basically alive for all of molecular biology as a field. They look like they're in electronics labs in this photo. That's actually very relevant because when you study the brain, you often have to have a lot of electrophysiology equipment, and I'm guessing these are all their like recording equipment. Very arcane kind of stuff. Where's the cats in the photo? You probably wouldn't want to see them. So they would be awarded the Nobel Prize for Physiology and Medicine in 1981 for their work involving the visual system, which means, yes, cats did win a Nobel Prize, which is very cool. Okay, so the question that Hubel and Wiesel wanted to answer is, how does visual stimuli affect the brain? So, rewind back to the 1950s, where our conceptual understanding of neuroscience was still in its infancy. We know there's an eye and we know that the eye is connected to the brain, but we don't really know anything beyond that in terms of how anything works. So say you have a very simple system here. You have an image that you are looking at. That image, the photons that bounce off of that image are hitting your retina, and that retina is sending a signal to your brain. How do you figure out how the eye is wired to that brain, specifically the visual cortex? And so I know this is kind of an open question, but I think it's fun. It's fun to try to trace the steps of these Nobel Prize winners, because oftentimes you can actually follow. You're all smart enough to retrace their steps. The difference between you, I say this all the time, the difference between you and a Nobel Prize winner is that they've just done a lot more science and done it at the right time. But you are also capable of doing this. So if you wanted to set up an experiment to try and see what the relationship between the eye and the brain is, what would you do? What would you do? What would you do? What kinds of experiments do you think? And don't worry if it's possible, not possible. I'll tell you whether or not the technology exists. This is just a fun little sandbox to play in to try to get the creative juices flowing. What would you do to try and see what the relationship between processing or receiving visual input from the eye is to the actual brain itself. Yeah. Okay, so we've got some suggestions here. So Marlon says naive approaches would probably be like electrode arrays, but that can be somewhat disruptive. Okay, so using an electrode, so something that can measure neuron activity. That's interesting. Leo said I probably apply voltage at a cell and probe the other side to see where it ends up. That is interesting too. So sort of trying to see if you can directly stimulate the retina itself or some part of the eye itself, and then seeing what happens on the other end. I like that format, but I will say applying voltage at one end, there's a more straightforward way to do that versus something that requires that level of manipulation. A guy says, first look at the anatomy, track what goes out of the eyes, and look where it goes. That's possible, and certainly something that they can do. I'm not super familiar with the state of technology back in the 50s, but I do have good reason to believe that going through the fine anatomy of the brain would be kind of difficult, especially once you start getting to synapses that are smaller and smaller and smaller. And of course, even if you take slices of the brain, you still have to process it, which can destroy these smaller connections. And of course, even if you were to have a good understanding of the general connections between different parts of the brains, doesn't necessarily mean that you know how things are connected on a functional level. So Guy, I think you're right in terms of just understanding the anatomy, and it's part of the fault of me wording the question that I worded it because it is so broad. So I'll say that your answer is correct, just not one that I was thinking of when I designed this question. Marlon says, show basic images to see how the brain responds, like an image on one side and white on the other. Yeah, okay, so this kind of is the equivalent of what Leo was saying, which is to stimulate the eye in some way. And of course, you could hook up an electrode hypothetically to the eye. I don't know how you would do it, but you hypothetically could. There's a much more easy way to stimulate the eye, and it's just to give the eye something to look at, right? I heard they fully mapped the brain of a fly recently. I believe so. There's like a fly brain atlas somewhere, which is very, very cool. Flywire? I don't remember what it's called. Fly Atlas, Flywire? I don't remember. Something like that. All right. So I will say that through a combination of what chat has been saying, I think we've successfully retraced what these scientists are going to try, which is very simple. Show an animal something and record it from different neurons. So you know roughly where the visual cortex is because you know that patients that have strokes in this part of the brain, they lose vision. Like you have a general understanding of where vision happens in the brain. So what you can do is you can stick an electrode in that area and show an animal an image and just see if you get a neuron spike, right? They're trying to deconvolute the connection between the eye and the actual brain. So show the animal an image, record from their visual cortex, and see what lights up and where, like what images light up what neurons, right? This is a very I'd say unbiased approach to understanding how the brain is mapping these out. Okay, so yeah, the way that you would measure these, you know, from an electrophysiological point is you can measure current just like electronics, you know. Right, so basically my idea the other way around. Yeah, exactly. You can probe either way. Okay, so what animal would you pick for this kind of experiment? So mice are always a worthwhile choice. In fact, this is an image that Iristroya just has because these experiments are so common in mice, right? Mice are incredible model organisms. There's even like a monument to mice as a model organism somewhere in Russia, I believe, to pay homage to the fact that mice are the premier mammalian research animal. But of course, you know, the idea of today's stream is that mice were not used for this experiment. But why are mice sort of the first animal you should think of, right? Well, they're a mammal and we are mammals. So if we want to study human health, then we should study a mammal. We're also not that distantly related. We're decently close cousins to rodents too, in terms of our evolutionary history. Mice breed really, really quick. I think when I was breeding mice, you leave mom and pop in a cage together. They'll probably do it the same day, and then you'll have a baby in, I want to say like 21 days, if I remember correctly. So very, very fast turnaround. They have large litters, right? So one breeding can yield, I think the highest I've seen is like maybe, gosh, like nine pups per mom. So you can generate a lot of research animals, and they are very cheap to house, feed, and entertain. Right? Again, I say animals don't have the best research lives, and that's true, but we do make sure that they are free from common indications of stress, right? So if mice are over grooming themselves, if they are fighting a lot, we have methods by which we make sure that they are calm at baseline, at least as calm as a research animal can be, right? They have dark hours, light hours. So I'm not saying that mice in the lab have the ideal life, but if you are in America, then there are standards, right? An unhealthy mouse is a bad research subject to begin with. So yeah, a little tangent right there. That's fast. Yeah, it is really, really fast. I mean, think of why rodents have been some of the more successful species to ever roam the planet, right? Rats and mice. They are really, really good breeders. All right. So, uh, there are two potential downsides to studying the visual system in a mouse, and there's also a third bonus answer that's not quite correct or not quite as relevant. So I want to take a little bit of time to just ask chat, why do you think a mouse might not be what these guys went for, right? Because again, if you are doing a mammal experiment, you should probably do it in a mouse unless you have a very, 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 very good reason to do so. Otherwise, just because there are so many advantages to using mice or rats even as your model organism. But Hubel and Wiesel went for the cat of all animals. Which, I mean, if you've ever handled a cat, you know that they are not easy to handle. Mice are easy to handle. So there are a lot of downsides to using cats too, but they felt like they could not go anywhere else besides the cat. So, what are your hypotheses? What are your hypotheses? Yeah, we've got a lot of good contributions in chat. Mice are really small. Yeah, that's certainly a factor. Do mice have any depth perception? Yeah, so maybe there's something different about the way that mice process information, visual information, right? Because remember, humans, we have front-facing eyes, right? We are omnivores on paper, but we are also visually styled after predators, right? We've got that forward-facing vision. Yeah, and Leo has brought that up because prey animals have a difference in the way that their visual systems are wired. Small lifespan. In terms of a mice, that's a benefit. You actually don't want an animal that lives too long because they're very, very expensive. Aren't mice omnivorous, meaning that they also hunt sometimes? That is true, but their eyes are more, I believe their eyes are more like an herbivore. I believe. I could be wrong on that, and I'll have to check later. They don't see color well. This is true, but cats also don't see color very well. In fact, humans are like goated vision seers. Of course, not as good as some birds, but like humans, pretty good on the color perception tier list. Yeah, yeah. So there are a lot of reasons. You all came up with great answers. So I'll reveal the primary ones, the primary reasons why we went for cats here. So as Marlon and others have said, mice have pretty small brains. Remember, we're in the 50s. We don't have like omega omega microscopes yet, right? Like we don't have these microscopes that can visualize things, without needing to fix tissue, fitting an electrode inside a mouse brain. I mean, it's possible, but it's very, very not that easy because of how small a mouse brain actually is. So not preferable, especially not back then when technology wasn't as good. Okay, the other thing is that rodent brains are not super similar in structure to human brains, right? So if you kind of look at these images of how the brains are structured, you can kind of see that rats are quite literally smooth-brained. And not only are they smooth-brained, but, in terms of the way things are organized, and you'll have to ask a real neuroscientist on how exactly they differ, but you can kind of look and already see. It's like the rat's got a different shaped brain, whereas the cat's just like ours, just a little different, right? A little different. Okay. This is the cat brain, right? So it looks enough like the human brain. And again, you might think, okay, well, maybe that's not a good enough reason. But again, we're back in the 50s. We got to pick an animal. And this seemed like a good choice at the time. Here's a bonus answer, which is not entirely what I would consider a major reason for avoiding rodents. Rodents have bad eyes, right? Human eyes are actually incredible. Um, even though we don't have like the resolution of like an eagle or something, we can perceive a lot of color. We have good vision tracking from our carnivorous, development. And from being an omnivore, we've had the investment into understanding, oh yeah, it's kind of important to know what color fruits are. So human eyes are really, really, really dang good. Cat eyes are also pretty amazing, but they're min-maxed a bit differently, right? These cat eyes, I don't know if you know this, but cats actually have trouble seeing things that are directly in front of them. They actually are farsighted. I think farsighted, right? They have better resolution further away than they do closer to them. And they're also min-maxed to detect motion, right? So, we're not really all that similar to cats, but our brains are similarly shaped, which is like not a bad start, right? So mice eyes, not super good. Mice would prefer to pee in the dark all the time. And this is, again, not a real reason why mice are worse, but it's something to note, right? Because again, back in the 50s, I don't think that this was really bothering them all that much because this is exploratory, right? Exploratory. Min-maxed, yeah, like evolved for. It's just my term for evolution, right? So cats don't really care about resolution for things that are very close to them, right? So things that are like 5 inches from their face are very blurry. So they're not like that good at focusing in that region. But things that are far away, like if a mouse is 20 feet away, they'll have very good vision of them. So yeah, so we're not super similar to either organism, but at least our brains are more similar in structure to cats than they are to mice. Yeah. All right. So here's another question. Why don't we just use a chimpanzee instead, right? If we're talking about similarities between different animal brains, why bother doing this research in cats? Why not just use a chimp? And I'm wanting to probe the audience in terms of why do you think they avoided using chimpanzees here? Why do you think? What do you think? Mice are molded by the darkness, true. But actually, their like vision is not that good. He's a Swedish researcher and didn't have the funding. I mean, you say that, but ethical concerns and reproduction rate. Um, there's always ethical concerns in working with any research animal, not necessarily any more or qualitatively more for a primate than any other research animal. Of course, you want to have every research animal live the best life they can under these circumstances. Harder to work with? True. Cats are notably finicky, if you've ever had a cat, but also chimpanzees are not necessarily harder to work with either. They can be trained and stuff. Whereas if you've tried training a cat, they are you can train cats, but they're not as known as being trainable. They're much more expensive and reproduce much slower. Yeah. So, uh, yeah, you pretty much nailed it on the head. They're too similar to humans. Maybe they needed to use cats for a better comparison. You would actually want something as close to human as possible. In an ideal world where you didn't have to care about ethics or money, but Will Not has hit the nail on the head. They're more expensive and they reproduce much slower. And so my follow-up questions here is, how much money do you think a single chimpanzee costs for their life in a research lab? Right? Because here's something that I see quite often on our favorite site to dunk on, Reddit. Common refrain on Reddit is, this research was done in mice, so it doesn't matter. This research was done in whatever animal. If it's not a primate or it's not a human, then we should just sleep on it. Who cares? To which I say, do you know how expensive it is to do research in one chimpanzee? Just one chimpanzee. One chimpanzee. Come on, get your guesses. This is a crazy number. Archie says way too much. Way too much. I mean, appropriate for the chimpanzee. You actually want people to spend a lot of money on housing a chimpanzee, right? Our evolutionary cousins, right? But that means that you have to have an excellent, an excellent reason to ever do something in a chimpanzee. A lifetime, a chimpanzee's lifetime. Let's get some more guesses in. At least 10 bananas per day. Oh, at least. I'm gonna guess like 40K a year is that minimum wage-ish. Let's see. I actually don't know, because I don't know the typical lifespan of a chimpanzee. So I took the figure that is the entire lifetime of a chimpanzee, not per year. Okay, let's reveal the answer. A lifetime for a single primate can be up to about $1 million for a lifetime care of a chimpanzee. $1 million for a single research primate. So of course, you would expect that if you are going to try and get statistically relevant ends, you would need to do a couple of primates, right? It is not uncommon to see research done in primates have ends of less than 5, because $1 million just for one primate. That's not even including like the broader lab budget. That's the part of the lab's budget that is allocated just to have this chimpanzee around, right? This is nuts, right? And to compare like the cost of a mouse, a cage of mice in my old lab was about $1 a day to maintain. So the lifetime of a mouse was about $760 because they live about 2 years. And you get like 5 of them per cage. So per mice, it's 760 divided by 5 for 2 years, right? That's crazy, right? I hope this makes it very clear. I'm sure in my audience, you all already know, like, yeah, sometimes you do science and you can't do it in a chimpanzee because you're going to go broke instantly and you might not even get something that makes any sense, right? So what is my source for this? It's Trust Me Bro. It's legacy knowledge. I worked in a lab that was next to, or rather I wasn't next to them, but I was 2 floors underneath the primate lab, and something that the animal facilitator told me was that a chimpanzee can cost like $1 million. And then I also found other sources for this, but they weren't like hard research papers, so I didn't bother to cite them. But this is like the general vibe, is that it's about $1 million for a chimpanzee. So what this means is that, of course, when you're on Reddit and are on R Science, and there are Redditors that are complaining about how this paper isn't perfect and it doesn't mean anything, just note that research is like a puzzle where you have to balance the motivation, the outcome that you are trying to get, the cost efficiency of your experimental system, and of course, ethics, right? You don't just do exploratory research in a chimpanzee. You usually have to do a lot of exploring before you can justify doing a chimpanzee. Where do these chimpanzees come from? I don't actually know. I was a mouse guy myself, so I don't actually know where chimpanzees for research are sourced from. But yeah, I wanted to drive home. I personally do not take animal research very lightly at all. Okay, so cats solve the issue that mice have, right? Cats share more cortical structure similarities to humans and mice do, so they are at least more anatomically similar to humans than mice are. Maybe not surprising. They also have relatively large and well-defined structures in their visual cortex, right? Again, cat eyes are really, really good. Cats rely a lot on their vision for a lot of things, even if the way that they see the world is different than ours, right? They're also, I think, missing a cone or something, right? Like we have tricolor vision. I think cats have like bicolor vision, right? So it's not a perfect match, but it's more similar. And, you know, Hubel and Wiesel felt like this was enough to justify going for research in the cat. They've got much larger brains, and so you can stick electrodes in different places and have it not be a total nightmare. Okay. Do extra mice get turned into snake food afterwards? For the most part, so there are some labs where you can adopt these research animals, which is super cute. You might check a local university to see if they have a program like that, if you want to allow a laboratory animal to retire peacefully. Otherwise, they are incinerated. Of course, after euthanasia. Okay, so Hubel and Wiesel, of course, we were saying they were going to hook up electrodes to the visual cortex of the cat and show them some stuff. In this case, they were looking at shapes, right? So they were showing these cats, different shapes, and what they were finding was that they were failing to get an actual response, right? This was very, very frustrating. It was very frustrating, right? So Hubel and Wiesel hooked electrodes up to the visual cortex in cat brains and showed the cats a series of shapes, hoping to find neurons that fired in response. But sticking, that electrode in the neurons one by one, showing them different slides, they're like, they're just not seeing anything. Nothing they did would produce an effect. It's almost as if our entire understanding of the visual system is wrong, right? Because this is incredibly depressing. Also, to surprise Pants's comment, before anybody says anything, Squid Game does not have a monopoly on shapes. Squid Game does not have a monopoly on shapes. Yeah, this was really, really weird, right? You show the cat visual stimuli and you're just not able to get a neuron to fire for some reason. And I feel this to like a very deep extent because I am a former neurobiologist, right? I feel this pain very, very deeply. I say, oh, just stick an electrode in there. But in reality, electrophysiology can be a huge pain. This is an electrophysiology setup. So Marlon had pointed out that there looked to be a lot of electronics equipment in that picture, that old picture of the Nobel Prize winners Hubel and Wiesel. I'm guessing that was their electrophysiology setup. And so I want to talk a little bit about this monster of the machine, right? This multi-million-dollar recording setup. This equipment is deadly expensive. It is omega sensitive. So even though I never did any recordings because I was more of a biologist than a neuroscientist, I know that that room had to be treated very, very carefully. Like there was a vibration blanket used to seal off that room from the rest of the lab so that people walking, to and from the lab wouldn't send shocks into the machine itself. It also had to be installed on a shock-proof table. Everything had to be grounded in a way where you could not have errant electrical current mess up your recording. Like this is an enormous pain. So if you are thinking about studying neuroscience and you want a guaranteed job, go be an electrophysiologist. Learn how to do these recordings. It's going to be miserable, but once you learn how to do it, people are going to hire you because nobody wants to do this kind of work. Nobody wants to sit and ground the instrument and test all the electrodes and make all the liquids to the exact specifications and troubleshoot this god-awful machine. If you're looking for work and you're doing research, electrophysiologists. You can thank me later, or you can curse me later, depending on how you feel about it. Yeah. All right. So, when you are doing these recordings, if you're not seeing a response, you're never 100% sure if nothing is happening because nothing is happening biologically, or something is wrong with your rig, right? Like if you can't get a reading off a neuron you think you should get a reading from, it could just be because you screwed up the reading somehow. And so I really feel for these two guys, probably sitting there probing this cat brain, showing them shapes, being like, oh God, do we have to take down the machine again and recalibrate everything and make sure it works? But luckily for them, and luckily for us, a Nobel Prize started with a slide that glitched. So I showed earlier, they were showing these cats pictures of shapes just to prod, they had to start with something. And in their slide progressor, an error, a mechanical error in the slide progressor and the projector itself caused the slide to get stuck. And what that caused was a large vertical shadow of the slide, like the edge of the slide casting a shadow on the actual projector. And so when the cat saw this long, dark, heavily defined line, that's when that neuron started firing. Ping, 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 like that neuron started firing. Da, 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 da. And you can imagine that these two scientists being like, oh my God, we're not getting anything. Do we have to take the machine down? All of a sudden the slide breaks and then they hit the jackpot. Ding, 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 ding. Right? That neuron is firing like crazy. Firing like crazy. All right. And so what happened there, right? A slide getting stuck was correlated with a neuron firing rapidly, right? At this stage, it's correlation. They first have to reproduce this before they can conclude anything causal. Were they measuring without changing the stimulus? They were showing them multiple shapes, right? Progressing slides, triangle, circle, square, whatever. But then when you got to this very heavy dark line, that's when things started really popping off. So Hubel and Wiesel realized that whatever neuron they were recording from was reacting to the shadow of the slide projected on the screen, a straight, dark line. Do they have to use a new cat every time? I'm not entirely sure how, the particulars of that setup. So this forms the basics of our visual processing. What they had just discovered by accident here is something called edge detection, right? Edge detection. So this is going to need a little bit of context. So a miraculous accident provided deep insight into how our visual information is actually processed. So Hubel and Wiesel were probing around what's called the V1 region of the visual cortex, right? They knew roughly where, they knew where the visual cortex was, but maybe they didn't have all the details as to what the different layers did. And they were probing around a region they knew was important for visual processing, but they didn't exactly understand why. So because they had found out that there was this one neuron that was responding very strongly to that one dark edge or a set of neurons, I'm not actually sure if they did like field recording or if they did a single cell recording. I don't actually know. But whatever it was, that region of neurons was firing like crazy to this one line. So, later on, they would really kind of tease apart what this would mean in terms of how visual stimulus is converted into information. So they didn't know it at the time, but this region is the first pass at interpreting visual information. So you take in those photons from your retina, but in order for you to perceive that image, your brain has to kind of parse it in a kind of way. And so these particular neurons that they were recording from in the V1 region of the visual cortex are the first pass neurons, right? These are the first neurons that receive that information and begin deconvoluting what the heck it is that you are seeing. So these particular neurons react to strong edges. They are line detectors, forming the first schema of what we're looking at. So if we have this picture of this labubu, those V1 neurons in that visual cortex, they're going to not look at the whole image, but they're going to look for, or they're going to react for very hard and dark, heavy lines, and then pass the information on, further into the brain, right? So that's kind of interesting. I want to hear what your reaction to that finding is. Did you know that visual interpretation happened in a layered kind of way, that you didn't just snap a picture, screenshot a picture, and then that's immediately what your brain recognizes, but your brain parses visual information layer by layer. And this first layer in this V1 cortex is edge detection. So how do you feel about that? How does that strike you? Is that interesting to you, or is it just me? And apparently, if you are like a computer scientist, this is the basis by which, machine vision occurs as well, I believe, where there's a similar layered schemata of first you want to detect edges and then you can fill in the details later. And I find that really fascinating. Like, evolution did a pretty bang up job on on making sure that we can see. I guess that's what makes depth perception. Got to see what's closest. I actually don't know how depth perception is constructed in the brain. I'm a fake neuroscientist. I'm a neurobiologist. But I wouldn't be surprised if depth perception was like one of the layers, or if it is parsed in a certain kind of way. I think depth perception is also, if I remember correctly, integrating information from both eyes and seeing how the vision differs from both eyes. Allows you to construct that image appropriately. It probably explains why false edges make camouflage so effective as opposed to just matching color. It might. That actually makes sense. It actually might. That is consistent with this idea that edge detection, if you trip up our edge detection immediately, whatever garbage is going to come out of that edge detection might not be interpretable. So very, very cool. So now you have a sandbox to play in. You have a sandbox, right? Because now, okay, I've mapped a line in that region, in that visual field, to this neuron. Now, what can you do? You can vary the line angles. You can vary the line position. Like, what if I put a line in the left part of the vision for the cat, or the right part of the vision for the cat? Or what if I change the angle? What if it's a horizontal line, et cetera, et cetera, et cetera? So Hubel and Wiesel did a lot of work just showing cats a bunch of lines and seeing which neurons lit up in response to those lines. So they're getting this idea that our vision starts off as interpreting line work, and now we're going to map what part of your visual field corresponds to what part of the brain. That to me is incredible, right? That in the 60s, 1960s, we just started molecular biology, right? Molecular biology is pretty new at this point. They're already being like, this line that is in this position of your visual field is sending signals to this part of your brain. And I think that's amazing. That's crazy, right? To know that there are pixels that you interpret in your retina, or you know, I say pixels, of course, your vision isn't really pixel. But if you imagine a computer screen, it's like understanding which pixels on your computer screen link up to the processor. That's the level of detail they're able to get just by being like, let's show the cat a horizontal line 2 inches from the bottom of the screen and find which neuron is firing. Of course, this is a simplification of what they actually did, but this is about how they constructed this understanding of the visual field. Yeah. All right. What happens if the cat sees a continuously rotating edge? You're getting into a topic that I was hoping to avoid, because motion is another thing that they worked on, but I purposely avoided here because I'm not smart enough to figure it out in the time it took for me to make this slide, right? So not only were they checking neurons for their ability to react to hard lines, but they were also finding interesting things about movement, right? If you moved the lines, that would cause neurons to fire as well. So yeah, a lot of information that they were able to glean because of a broken slide projector. It's kind of crazy. A Nobel Prize completely by accident. Honestly, I don't know how these people figure things this detailed out with neurons and all. Say more about that, Amarilla. So what part of the equation is missing for you? Because I actually think it's really interesting to analyze the way that these people think, because insight is different, right? Insight, it's not the things that Nobel Prize winning scientists do are not complicated to understand at a fundamental level, right? You have a brain, I have a brain. We all evolved over hundreds of millions of years to be pretty dang smart. The difference is that the Nobel Prize winner had insight, that they were able to come up with the idea before anybody else. So I would never have claimed to be like, you know, I could have done that. I guess hypothetically I could have done that, but I didn't. And the difference is that they had insight, I did not, right? Very interesting. So from this careful tracing of visual field stimulus to the visual cortex, they were able to discover a structure within the visual cortex called ocular dominance columns, which I think are really cool. So this is what this looks like. Of course, we have this information now, but inputs from each eye are segregated in certain mammals into columns by the lateral geniculate nucleus, or LGN. So let's take a look up here. You have the left eye, and the left eye is sending inputs down to the LGN, and that LGN is going to sort out these connections to this particular region of the brain, and then these layers from the lateral geniculate nucleus are going to get fed in a structure towards the back of the visual cortex called ocular dominance columns, which, as they might imply, are like columns of cells. So you can kind of see by this color matching that in this series of ocular dominance columns, you have certain columns that originate from the left eye, right? If you trace this red, you can see that from this red left eye, you have these pink colored columns. So these are regions of the brain that map to the left eye. And then in between these pink columns, you have these blue columns. These columns are wired from the right eye, right? So of course we know this now. Back then they were still working this out, which is really cool. So you might be familiar with this concept that like the left brain controls the right side. This is similar to that idea, but not the same, right? In your brain, both of your eyes are sending inputs to both sides of your brain. So it's not like left eye goes to right brain or left eye goes to left brain. Left eye is sending specific inputs into your right brain and your left brain. And the way that these are intermingled is through these interlaced columns, which are very neat. I've had a pretty hard time myself visualizing this, so I found a really interesting picture online, which of course I can't show you because it is copyrighted. So boo, copyright. I hate copyright. This is science. It should just all be online for free because taxpayer dollar and everything like that. But here's my best attempt at drawing it, and then I'll show you how to find this real image on your own. So if you isolate the V1 visual cortex and you get an animal to close their eye, you can change the metabolic activity, right? So in this part of the brain, if you have one eye closed, like half of that portion of the brain just shuts down. White here represents recent metabolic activity. So this is glucose being chewed up for energy. And then black represents no metabolic activity. So the black stripes are regions of the brain where it is quiet, which I find really, really cool. So these are literally ocular dominance columns. They are columns in the brain that either all light up or all shut off, depending on which eye's turned on. So if you want to find this real image, Google image ocular dominance columns, and you can find the real brain image with glucose imaging, and it's awesome. Do it now if you can, and look for the zebra stripes. I'll take a little bit of a water break so you can look at this for yourself, right? And look for this, an image that looks like this, right? Glucose metabolism in the brain, specifically the ocular dominance columns. So let me know when you have successfully looked it up for yourself, because I refuse to move on unless at least some of you see this part of the brain for yourself, because my dumb little drawing isn't going to do it justice. Yeah. Ocular dominance columns. And it's so cool to see such a like complicated idea as vision be distilled into biology, right? Because you know, a microscope can capture an image, right? It's just photons filtering through lenses or whatever. But in terms of like, how does your brain reliably interpret information? To see it be simplified, of course, simplified to a certain degree as a matter of biology is really, really striking. I love that the brain stays consistent with the wrong side idea by putting the visual center at the very back of the brain. Yeah, it is a little counterintuitive, isn't it? It looks like zebra print. It does look like zebra print. It does look like zebra print. To be honest, the lamellar structure reminds me of Turing patterns to a certain extent. Yeah, you can see a lot of different patterns in that. To me, it just looked like zebra stripes, but they do look quite different, especially from different angles, right? Because you can look at it from the top too, but from the side, they are columnar, right? Columns up and down, up and down. Also, there's a bunch of other like weird visual things in terms of like your retina is turning things upside down in weird ways. I don't fully understand those visual processing things myself, or rather, it's been a while since I thought about it. Or like the retina turns it upside down and then your brain deconvolutes it. Like I don't remember. There's a whole bunch of steps by which visual input is processed. And it's all very, very cool. And the fact that you can get a read on which part of the brains light up depending on which eye is opened or closed is like absolutely wild. It'd be nuts. All right. So you can read more about ocular dominance columns in this blog post. It's in the description. I link this because I found it to be a very easy read, and you also get to learn about the third Nobel Prize winner that did research in cats and the eyes, but I didn't have time to cover them. I didn't have time to like vet this blog post. I just generally trust Psychology Today as a general source. Generally, there are exceptions. But if you want to learn more, I think it's okay to read that. It's just I'm reluctant to share information I myself have not vetted. Let me just put it that way. Upside down retina is just the lenses inverting the image. Ah, okay. So that is like their primary claim to fame, but they did a lot more with cats and visual processing. Of course, all of this research accumulates in their Nobel Prize win before. It's a pet peeve of mine when people are like, how does your brain know to turn the image around? Yeah, my bad. I did hedge a lot that I didn't quite remember how it worked. Yeah. Okay, so that was like the neuroscience part, the part that I'm very uncomfortable with, but now I want to talk about plasticity, which is in the realm of cells, which I am much more comfortable talking about, right? This is cell stuff now, plasticity. So what is plasticity? So plasticity is a general characteristic. You can call a whole bunch of things plastic, but if you are calling things plastic in a scientific sense as a descriptor, not referring to the material it's made out of, it means it is a general characteristic that indicates the ability to change, right? So I worked on mechanisms of synaptic plasticity in graduate school, so this is near and dear to my heart. So this is referring to the fact that neurons have the ability to change the strength by which they detect signal and the strength by which they send out signal. They are tunable, right? They can get a little louder, they can get a little quieter, they can hear a bit better, they can hear a bit worse, right? They are capable of changing that connection. So cats have contributed immensely to our understanding of how the visual system develops through plasticity, which is really, really exciting to me. So yeah, part of the reason why I wanted to do this stream is because my research experience probably would not have been possible without the contribution of cats, which is very, very cool. Okay, so plasticity has been hypothesized for hundreds of years before this research done in cats, right? So this idea that neurons are plastic is not new. We all do in fact learn, after all, and if you want to believe that everything living has a biological basis instead of just well, I guess I should be careful with my terminology here, right? If you want to believe that learning is reflected in your biology versus purely in the realm of psychology, then it is not unusual to think of neurons as being plastic. I know it's not a very clear boundary, right? Because it's not like psychology ignores the idea of neurons, right? Of course, they work with the brain. They know about neurons. But if you want to keep learning as a completely psychological phenomenon, like completely within the mind, that would be wrong, right? Because there's a biological basis behind the mind. And I recently had a conversation that was interesting to me where this is a bit of a tangent, but like we really, I think, should be thinking of the mind as separate from the biology of the brain. Maybe not completely separate, but separate enough, because your mind, the thing that is actively perceiving and thinking, is unusual. It's special and independent and bizarre and infinitely complex, but underneath the mind is a biological basis. And those two things don't have like a worked out connection that is very, very solid. So if you accept the idea that learning has to be reflected in biology instead of just a change in this vague concept of the mind, then you have to conclude that neurons have to change somehow. Maybe you get more neurons, maybe you get less neurons, maybe the neurons are better, maybe the neurons are worse, right? So this idea is hundreds of years old. Almost as old as neurons, I'm guessing. So if learning is reflected as a physical process or has physical components to it, then it might be changing, right? It might as well be changing of neurons somehow. So Hebbian plasticity is a type of plasticity that Marlin referred to, which is a concept that states that neurons who fire together wire together. And so you could see this as this presynaptic neuron, neuron A. When this fires, if this neuron also fires, then these two will strengthen their bond, right? They will reinforce their connection. And I say that very, very, very carefully because I think I had this misconception too when I was studying synaptic plasticity for a long time, and then it finally clicked. Neurons that fire together, wire together is not driven all the time, right? Neurons who fire together, wire together is a correlation. And what do I mean by that? I mean that if neuron A is firing a lot and neuron B is firing a moderate amount, if these two happen to be firing at the same time, then their connection will strengthen, right? This is literally neurons who fire together, as in neurons who fire one after another, not because of anything, but just happen to be firing one after another, will strengthen their bond. That's an important distinction, not for this presentation, but it's a distinction that has been hammered into my head for my entire time in graduate school. So if a postsynaptic neuron fires, postsynaptic neuron, this guy right here, if this guy fires an action potential right after this guy fires an action potential, their connection will strengthen. Does that kind of make sense? I think that's a really important distinction that, at least for me, was a big misconception that, oh, neurons who fire together wire together. That means this neuron is forcing this neuron to fire, which is true to some degree. But the base principle is if these two neurons happen to be firing together, then they will strengthen their connection. Yeah, so that is the general concept of plasticity, right? And you can kind of extrapolate this to your own life, right? If you are studying, then those neurons that are corresponding to reinforcing the knowledge of a thing that you're studying, they will fire repeatedly. And the more you study, the more those neurons will fire, the stronger those connections between those neurons get, the better you'll be able to recall, the better you'll be able to apply the knowledge that you learn, et cetera, et cetera, et cetera. So, yeah, learning is a thing, and it happens biologically. So, the part that wasn't obvious, right? This idea of plasticity was obvious, or at least commonly, not like an outrageous thing to believe. But what wasn't obvious is that this idea of plasticity also governs the development of the visual system. And I think, in the year of our Lord 2025, this is not surprising. If you grew up in this era of biology, duh, right? Yeah, your eyes need to learn how to exist. That's essentially what this statement is. But back then, it wasn't that crazy to believe that your eyes just loaded in. They just, you loaded the program for the eyeball and it just worked, right? That's not that crazy to believe. I don't even think it's that crazy to believe now, like logically, right? So up until the 1960s and 70s, it wouldn't have been crazy to think that our eyes and brains just had genetic code that set everything up correctly, independent of your lived experience. Like simple lines of code, right? Initialize brain, initialize eyeball. Okay, those programs worked. The eye will develop fine. The brain will develop fine. That is not true, as it turns out. Correct wiring of the eye to the brain requires there to be input. In order for the eye to be correctly attached to the brain, you need to feed the eye information. You need to see, right? Especially at the beginning of your life, you need to actually use those eyes in order for the wiring to be set up correctly. Hebbian theory is apparently from 1949, so apparently general relativity was more obvious than neuroplasticity. That is true. I think Hebbian plasticity is what ends up being mostly correct, basically. But before that, people were hypothesizing different mechanisms. But neurons that wire together, fire together, caught fire. It was, it's a banger statement, and it happened to be true. And it remains true in certain contexts to this day. So does it kind of make sense, chat, why I find this so incredible, right? You really have to put yourself or practice putting yourself in a place of ignorance in order to draw as much joy as I do when I read these papers, right? Because, why would you believe that you needed to see in order for your eyes to work? It's obvious now that we learned how neurons worked, but back then when we didn't really have too clear of an idea, why not just believe that your eyes just worked right out of the box, right? No, turns out they need to be trained. What does this have to do with cats? All of this research was done in cats. All of this research, the whole reason why we understand how the eye is connected to the brain is because of cats. They were incredible contributions to the world of understanding our visual centers. So it's obvious nowadays, this is basically the use it or lose it principle, but before a rigorous understanding of plasticity, this was not obvious. This is not obvious. So you be the scientist. This is another part where I want us all to hypothesize about what this is. So how would you test the hypothesis that the visual cortex needs visual input to develop normally? What experiment would you run here? What experiments would you run here? What experiments would you do to test whether or not you need visual input to develop normally? So does the V1 area in blind people, or cats for that matter, not have those stripes? I'm sure the answer is out there. I don't want to say anything because I did not read that particular experiment. But I wouldn't be surprised if their stripes were differently shaped, or if some of the stripes were wider or thinner, Leo. That's my hypothesis. I don't want to say that that's true because I didn't read it for myself. Yeah, Any blind cats in the room? Yeah. Cover their eyes. Okay. Blindfold. Baby mice. Again, we want to try to avoid using mice at this time period just because mice are like really hard to work with. Their brains are too small. Cover one eye for the animal's life and then see if they react to stimulation. Cut the inputs in both eyes or just one. Cover the eyes of newborn kittens. Grow kittens in complete darkness. So the correct answer here is a combination of what everybody is saying here. I'll show you what they actually went for, but again, a lot of your answers are correct in one form or another. And Wilnot's suggestion of growing kittens in complete darkness, they did do that, and they found that their vision was incredibly broken. But that's not the experiment that I want to talk about here. So, this is an experiment called monocular sensory deprivation. And so what you do is you have one normal eye of the cat, and then you sew the other eye shut. And again, laboratory animals do not live very happy lives, although we try our best. And you should know, and I say this later, but I want to get this out immediately, suturing one eye shut, we don't have to do that anymore. There are other ways to block off input, including just wearing an eye patch, right? But for them, they did the suture. So what this does is we already know from the previous work that both eyes are sending inputs to both ends of the visual cortex. So what they did is that this suturing of the eye shut closes one eye of the developing cat. You are depriving the cat of visual information from one of the eyeballs. That's not ethical. Ethics is relative, right? In order to understand the developing visual system, it was determined back then when these experiments were done that this is a worthwhile trade-off. Also, I'll talk about how this research has contributed to our understanding of medicine later on. It is kind of messed up. And again, I don't want to shy away from the fact that laboratory animals live hard lives. But again, we don't got to do this no more. We have eye patches. We have eye patches. And I also recommend that you don't go into biology if you're uncomfortable with inflicting harm on animals, because a lot of, I mean, you can if you do like bacteria and stuff, but if you want to do human health stuff, you are going to either be reading research that has misery inflicted onto animals, or you are going to be doing it yourself. So if you want to do like infectious disease, that might be easier because then you just work in bacteria or viruses or something. But yeah, progress has a price, and I never want my audience to forget that, right? Progress has a price. Because we now know that eyes send inputs to both hemispheres, we have a map of which inputs go where. We can see whether optical input is required for normal development, right? So because we know how the eyes are wired to the brain, thanks to these experiments, these Nobel Prize winning experiments, we can see if those parts of the brain get screwed up by having one eye shut. So things can change physiologically as well as neurologically, right? So you can have changes that are made to the brain structure. You can also have ways that change the brain in terms of like individual neurons that are not as obvious on a gross anatomical level. So what do you think happened next when these researchers blocked the eye of the cat and looked at their visual cortex, so the part of their brain that decodes this information? What do you suspect happened in those regions of the brain? This is a bit of a tougher question. Also, those experiments have been useful. Monocular sensory deprivation can be used to reduce neurological misconfiguration due to some ocular problems. So Guy, I'm actually going to address what you are talking about later on in the PowerPoint. So it is an interesting point, right? This research is really, really important for our understanding of the brain, also for medicine itself, right? It's smaller and deteriorated? In what way? In what way? In what way? If you could be a little more specific. We know that both eyes feed information to both sides of the brain. What do you think is the difference between the parts of the brain that were wired to the open eye versus the parts of the brain that were wired to the closed eye? What do you hypothesize happened? Fewer neurons and they're not as connected? Maybe, maybe. For sure, maybe. Any other guesses? How does the parts of the brain that are wired to the closed eye different than the parts of the eye that are wired to the open eye? I would imagine that the functional eye gets to gobble up the neurons which the closed eye isn't using. Interesting. Interesting points, right? This idea of compensation, right? Compensation. It's a good hypothesis. It's a good hypothesis. So, what did these scientists end up finding? So again, this is my sanity note. I should have added this before. Eye suture is not the only method of monocular deprivation anymore. We can do other things now. Here's what they found. The deprived eye had normal light reflexes. So they unsutured the eye, removed the sutures, tested the eye itself for functionality, shined a light in there, measured the pupil dilation reflex, found that the reflex was normal. Are you surprised? Is this surprising? What's your read on this? Are you surprised that after suturing the eye shut for a month in a newborn, the eye itself appears to have normal light reflexes? And does that surprise you? Does that surprise you? I thought it would be useless, the eye itself. Well, it's interesting, right? That's kind of crazy. My eyes hurt after waking up. Yeah, that must have been a crazy shock to the system for the cat. I'm not sure. Yeah, it's something interesting that they noted, and this is a mark of a truly good scientist, right? I didn't even think to check the eye for functionality. I was just thinking about checking the brain, but the eye was kind of normal, right? Kind of normal, and I don't know if that's surprising to any of you. I'm afraid I don't know enough about reflexes to know whether or not to be surprised. Yeah, so we're talking about reflexes. So reflexes are something that if you are in biology, you should be pretty familiar with, even if this region of the brain is not necessarily the same as the rest of the body. It works on similar principles, right? So reflexes, at least, in your limbs, these are neurons that are wired to the spinal cord, and they don't have to, a reflex happens in your limbs because you get a sensory sensation touching a sensitive nerve or whatever, and a reflex pushes the information around the spinal cord back to that arm. So you get the feeling, but then the immediate reaction doesn't have to go all the way back to the brain. It just happens, right? So reflexes generally don't require an input into the brain. So is it surprising that the deprived eye had normal reflexes? It's interesting, right? The reflex being okay is not something I'd expect, to be honest. Yeah, it does somewhat surprise me. It is cool, isn't it? But it is surprising and also unsurprising at the same time. We're talking about the eye getting wired to the brain. It's maybe not that surprising that the eye itself is still okay, right? Because the eye is just an organ that is used to detect light, right? It's not the thing that is learning. The brain is the thing that is learning and putting together information. So maybe it's surprising, maybe it's not surprising. But this gave us an additional insight into the fact that the eye itself is not the thing that is doing the learning. And today that's more obvious. Back then, not obvious. So I think this was a very, very good thing to test. Very, very good thing to test. So the eye is already formed at birth, but the neuron connections need stimulation to develop. Exactly. Or that's it's not entirely true. It's more like the neuron connections need stimulation to survive, to avoid being trimmed. So vision was poor in the deprived eye. Cats could not perceive objects in the deprived eye, right? You close the other eye, give the cat an eye patch over their good eye, and they are bumping into everything, right? They can't figure out what the heck it is that they are looking at when they are using the closed eye. Yeah. The deprived eye could not evoke nearly as many cortical responses as the normal eye. So when you open and close one eye at a time and you show the cat visual stimuli, the open eye, they stick that electrode in the brain, bang, 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 bang. Those neurons are pinging like crazy. And then when you open the eye that was shut down for the first month of their life, you try to measure from that region of the brain corresponding to the closed eye, and they just don't evoke action potentials, right? They are not pinging nearly as often as the open eye was. Remember, kittens are born blind. They're born blind, but they're not born not receiving input information. Or was that kind of your point, Alexandros? That might have been. We might be in agreement. So, cortical regions corresponding to the deprived eye atrophied, right? So some of y'all have said this in chat, the parts of the brain that were connected to the shut eye shrunk, they atrophied. And then the cortical regions corresponding to the open eye enlarged, right? So it looked like there's this compensation effect, that the eye that was open for the entire life of the cat was overtaking the regions where the deprived region shrunk away. So there was compensation. Now, whether or not that's because there were more neurons, fewer neurons, more synapses, fewer synapses, I don't actually have that answer. The answer is out there somewhere. I didn't put it in the slide because I couldn't, I didn't find the exact answer in time. But from what they saw, one region was bigger, one region was smaller. Is it because those neurons had more or less connections, or were there more or fewer neurons? That I don't think they knew at that particular point. So this effect was strongest in newborns, right? If a newborn kitten has their eye sutured or has an eye patch put on top of the eye, this blinding effect was strongest, right? If you raise the kitten normally for a month and then suture the eye or give them an eye patch, they're a lot better off. Not completely normal, but a lot better off. So it seems like there is this critical period. Actually, let me not say that. Let me not spoil the next slide. These are the observations, right? I made a deliberate effort because, if you're new to my stream, I like to try to cultivate scientific reasoning in my audience, right? I don't want to just spoon feed you the answers. That's why I ask you all these questions. It's not because there's a pop quiz. It is because by forcing yourself to answer, even if you're wrong, but by forcing yourself to try and put yourself in the shoes of these scientists, I think you can improve your understanding of how to determine truth, at least in the experimental world of science. So these are not interpretations. These are just the data that the authors had collected. And now we're going to do an exercise. On one column, I have the observation. And in the next column, we're going to practice drawing some conclusions, right? Drawing some conclusions. Do they test if vision improved once the eyes are opened? Sorry for jumping ahead. I believe once those kittens went blind in that eye, they were pretty much done for. They were always going to be blind in that eye if you do it from being a newborn. So I will do the first, or actually, no, you can do the first one. This one's not too bad. So what is the conclusion that we can draw if we observe the light reflexes are normal in the deprived eye? What do you conclude? What is your conclusion? In absence of any other result, you see that the eye reflexes are normal. What do you conclude? And the reason why I like doing this exercise is because people on Reddit, my favorite community to dunk on, they misinterpret result from conclusion all the dang time, right? There is the thing that the data says, which is the result, and then there's your interpretation, which can be different or more far-reaching or incorrect, right? The data are the data. The data are, as long as it was collected correctly, the data are quote-unquote correct. It's your interpretation that can be wrong or right? So, what is your conclusion here? Reflexes are hard baked. Yeah, you could conclude that reflexes are hard-coded in some kind of way, that they do not rely on some of this other gooey plasticity. Reflexes aren't plastic and are actually formed before birth. You could conclude that. You could. Reflective nerves can't be trimmed away as easily, or aren't trimmed, period. At least from what they are experimenting with, yeah? That the eyes can see light from a closed eyelid. This is a closed eyelid that has been closed for a full month after the cat was born and then reopened. And so when you say see, it's kind of, seeing happens in your brain, right? The eye is just the lens that collects the light. Evolutionarily speaking, it seems that the eye reflexes are necessary to be autonomic from birth to protect the eyes and help the organism reach maturity. Yeah, I mean, that is drawing a conclusion, yeah? That you could conclude from an evolutionary perspective that makes sense, that would be disconnected, that it would be too dangerous to have reflexes have to be learned, that it should be hard-coded. That reflexes don't entirely rely on eye health. That's an interesting interpretation. and maybe is an example of like our limitation in language. Here's what I would conclude here. The eye itself is still somewhat functional. I won't say it's seeing, because again, seeing is a thing that your brain does, but the mechanics of the eye are normal. So what I'm concluding here is that if the cat is blind, that the dysfunction might be happening in the brain or somewhere along the route to the brain. That the dysfunction in the blind eye is not because the eye cannot receive light, right? Maybe the retina is screwed up, but at the very least, like the iris, the sclera, or whatever. I'm not an eye scientist, but like the moving parts of the eye that's supposed to do its job, which is to collect light, that's probably still pretty functional, right? That's my conclusion, right? I'm not saying your conclusions are wrong, but in terms of like the breadcrumbs to their final conclusion, this is like the path that I would take, right? The eye itself is somewhat functional, and extra credit is the stuff that chat said, right? Reflexes are hard baked. Reflexes don't rely on plasticity. Those are all very valid, but building up to their final conclusions, this is the route that we are walking towards. So the second observation they made was vision was poor in the deprived eye. Now we're talking about sight, right? Light reflexes, that's not sight. That's just do the thing that the eye was built to do, which is to collect light. And it did look like the eye was collecting light, so whatever broken vision is not because the eye failed to collect light. Does that kind of make sense? This is so that they have a hypothesis that is ruled out, right? Because you could say, okay, you close the cat's eye for a month right after birth, and the cat went blind in that eye. You could conclude you damaged the eye. But here they're showing that the eye reflexes are mostly normal, which means that they probably didn't damage the eye, right? In the suturing, like maybe the suture got infected and then it damaged the cornea or whatever. That does not appear to be the case because the reflexes are still functional. Now, is it a perfect observation? Did they test every parameter, intraocular pressure, all of that good stuff? No. They did a broad assessment of the health of the eye and concluded that, yeah, it still reacts to light, so it's probably pretty chill. What about this next conclusion? Even when the eye is collecting light, the cat can't see. It's blind in that eye. So what do you conclude there? What do you conclude there? What is your conclusion? The eye itself, let's just assume. I know I talked a big deal on hedging whether or not the eye was actually functional, but let's just assume we are following this breadcrumb trail. The eye is working, it can collect light. The cat is blind. Why is the cat blind? Why is the cat blind in that eye? Kind of interesting, right? Kind of an interesting question. Why is the cat blind? He says something else isn't working. Yes, something else isn't working. If the eye is fine, what else could it be? What else could it be? If you had to guess, what would it be? Somewhere between eye and mind, there's a break in the info flow. Yeah. Somewhere in between the organ that collects light and the organ that tells whatever thing your consciousness is, something along that tract is broken or broke. Exactly. So we're narrowing down on a story here. If the eye is functional, then dysfunction might be in the brain somewhere. That's what I would conclude. Or along the brain. That's also fine too. That's actually even better. I like the way that Alexandros said it. Somewhere between the eye and mind. That's a little more broad, whereas my hypothesis is a bit more pointed. But because my hypothesis is a bit more pointed, it can lose out on the fact that maybe it's the connection between eye and brain that is weakening instead of something in the brain. The cat was paralyzed in its eye, or could they move them? I believe the eye movement was normal as well, but kind of hard to assess because they can't see in that eye, right? So it's not like they can track using only that eye. But I think if I were to guess, if you showed them an object, that the eye would move accordingly, or maybe not. Or maybe not. Maybe that eye has problems tracking. That's actually an interesting question. I don't want to conclude too much because I don't remember from reading whether or not the movement of the eye was normal, but the reflexes were normal. Yeah, so the eye can't communicate with the brain for some reason. Yeah, exactly. And then so I like that level of broadness. That's probably what I would put here instead, but because I wrote this in, we're just going to go with that train of thought. So if the dysfunction is happening in the brain, okay, then we test for that. So they took their electrode, they stuck it into the visual cortex and measured activity from the brain region that's hooked up to the deprived eye, and they found that those neurons were not firing in response to visual input. So what would you conclude there? What would you conclude there? You stuck an electrode in the visual part of the brain that corresponds to the eye that did not get input for the first couple months of their life, and those neurons are not firing. So what would you conclude there? Cat's blind AF. That is true. That cat is really blind. But what does it say about the brain where you are not getting that neuron activity when you stick an electrode there? What might you conclude? And hopefully you can kind of see that the conclusions, they connect to the observations, right? The eye itself is normal is the first conclusion. The second conclusion is that the brain might be abnormal. You check the brain for abnormality. What do you conclude? Cat brain not working, but cat eye still working. Yeah. The visual cortex is damaged / dysfunctional in some way. Exactly, Leo. That is exactly where my brain would go next. Something is broken in the visual cortex. Neuron skill issue. Exactly. If dysfunction is in the brain, then maybe cortical regions corresponding to the eye have atrophied or are damaged in some kind of way. Right? This is a pointed hypothesis. You kind of need one to test, right? So, see how there's a logical line that starts with the first observation and there's a through line that continues. There's a very satisfying story here. The eye itself is normal, so we suspect that there's something wrong in the brain. We measure vision in the brain. Hey, we have vision abnormalities in the brain. Let's check for damage. Let's check for atrophy. Right? Let's check for something that explains why we can't get a response from this area of the visual cortex. So what did they find? The cortical region corresponding to the deprived eye atrophied. So what do you conclude there? What do you conclude there? The regions that are corresponding to the eye that was covered for the entire life of the cat are mostly gone. What's your conclusion there? What happened? How would you connect a lack of visual input to this brain damage? How would you try to explain it? What's going on here? There's damage in the brain corresponding to the deprived eye. That is the observation. What do you conclude? This is also like my students' least favorite activity, but I do like drag it out of them because you need to be able to take data and then follow the data. This is a useful exercise for following data. This may be useful to conducting visual input. That is true. Cortical regions, you find some cortical regions shrinking, you can probably conclude that those were important for that eye. Marlon says eye stimulus is necessary for visual cortical survival. Yeah, I'd say that that's not a bad conclusion to draw, that you need something pinging in your eyeball or pinging from your eyeball to keep those regions of the brain, we'll call it alive. Atrophy is a very broad term. It just means that like something that was there is no longer there, whether or not the neurons died or failed to do whatever. That's a little more specific. So eye stimulus is necessary for visual cortical maintenance, might be a little more fitting because they don't know that it's death yet. In fact, I don't know if it's death yet. Yeah, cortical regions need input to develop properly. Yeah, that is a good conclusion. So, if cortical regions corresponding to the deprived eye are atrophied, what about the open eye? So I forgot to put a conclusion here. Yeah, I like all of your conclusions, right? You need visual input to prevent this atrophy in the deprived eye. And then this leads to the next observation. If this region is atrophied, what about the other region corresponding to the open eye? Well, they find that they are enlarged. So it looks like there is some usage-based plasticity in visual development after birth. And then the next question would be, is there a critical period of plasticity, right? Like they saw, hey, look, this eye thing, right? It looks like you need the eye to be doing something for the brain to develop correctly. Okay, when does that end? If any, right? If you take a 25-year-old man and give him a permanent eye patch, do they get brain damage, et cetera, et cetera? And so when they tested this, they found that this effect was strongest in newborns. So they conclude that there is a critical period of visual plasticity. That's crazy, right? That is crazy. And you can kind of see, I don't know, I think this is worth a Nobel Prize, right? We went from being like, I don't know how the neurons on the eye and the neurons in the brain are connected. We got from like screwing around, poking in the brain, being like, what neurons are doing what? We went from that to finding out not only how the brain is structured in terms of inputs from both eyes, but also finding out that your eyes need to work in order for your brain to develop correctly. And this was all in the span of like 2 decades. And all of this research is thanks to the cat, right? All of this understanding is thanks to the cat. So again, we have to keep that in mind. So of course, I find this to be a discovery completely worthy of a Nobel Prize. There was a third guy who won the Nobel along with these guys. He also worked in cats. I didn't read about him because I didn't care. It's probably also pretty cool, but I just wanted to focus on these two for now. The focus of today's presentation is about the disentanglement of circuits that connect the eyes to the brain. That's what I wanted to talk about because it was a factor in my research in graduate school. While ocular dominance columns are not universal, they are consistent across felines and humans, so you don't actually need ocular dominance columns as a structure in your brain, surprisingly, right? A discovery worth of animal abuse charges. Again, progress has a price, right? Progress has a price. And again, we also don't need to do the suturing anymore. Could it have been done more humanely? With retrospect, yeah. As we improve technology, we can do things a lot more humanely these days, including using an eye patch, right? You can just have an opaque cup that you attach surgically and then can remove surgically, which might be a little more comfortable for the cat in question. But again, without their contributions, we would have been delayed at least 30 years, I think, in our understanding of how the visual system works. Okay, so Hubel and Wiesel taught us about what neurons are triggered in the cortex by the eyes, what inputs and where those inputs have to be, and a lot about how the developing nervous system ensures our visual system develops correctly, right? This is their contribution that won them the Nobel Prize. So, the real impact of this cat research, and of course, this is my cat, this is Patrikios. You hear her occasionally on stream singing a sad song. She's very vocal. And Patrikios shares a species with a Nobel Prize organism of 1981, right? Very, very significant, very, very good cat. The cat was fundamental to our fundamental understanding of visual development and also mechanisms of plasticity in our brains, right? It isn't an exaggeration to say that cats play a big role as to why we're able to understand what we understand in the brain. This is not just limited to the eyes, right? While the influence of this prize can be felt all over neurology and neuroscience and optometry, the most direct effect comes from treating lazy eye. So this is something that Guy had pointed to early on in stream. Coming back to mice, if I have an amazing sense of smell and hearing and I'm prey to virtually everything that moves, I'm probably fine with only a motion detector. Exactly. Something that is barreling towards you. All you have to know is you need to run. But yeah, like plasticity in the brain, right? Learning, LTP, LTD, all that good stuff. You now have a way to reliably induce or test or measure or cause learning because you know that the eye causes the brain to learn. You don't have to teach a mouse to do fancy tricks or whatever. You can do the deprivation. You can block off an eye. You don't even have to do it for a whole month. You don't have to cause the cat to go blind, right? If you want to test how certain neurons change their firing strength, you can deprive the cat for a day and see what happens when they don't get input for a day, and see what their neurons are like afterwards, right? Like, this not only had huge impacts on how we understand the visual system, but how we can test for this idea that neurons are learning to begin with. Like, this is a huge kind of discovery. Very, very important. Yeah, but like directly, if I had to point a direct impact beyond like the whole field as a thing, it would have to be lazy eye. Brikinji, my love. Hey, Mylan, welcome. So that's why some of my classmates had eye patches. Yes, that is exactly the condition we'll be talking about right now. So lazy eye, or can't say this, amblyopia, occurs when the brain favors one eye over the other. This can happen for several different reasons that I'm not qualified to talk about. Amblyopia often starts at the age of 5. Once that critical period closes, that eye will struggle to be very useful. So actually, people with this condition function fine, and then there's some reason, some offset in the eye, the way that it's connected to the brain. For one reason or another, one of those eyes gets weaker. And we already talked about neurons that wire together, fire together. The opposite side of that, neurons who do not fire together, will weaken their wiring together, right? So if you have an eye that gets a little bit lazy, it's just not quite as good. Over time, the ability to use that eye will decrease until you have severe vision defects in that one eye, right? So this can be quite devastating, right, in terms of shutting off half of your visual sense. Asterisk, right? Eyes are complicated. I simplified it just there. But in any case, like, this is a source for a decrease in quality of life for a lot of people. Before Hubel and Wiesel, it was commonly believed that amblyopia was a disease of the eye itself and not the connection to the brain. This is actually not that crazy, right? People with lazy eye often do have slight defects in the way that the eye functions, and that can compound into a blindness that happens in the brain. But it cannot explain everything about this condition. So this is unsurprising, right? This other wrong hypothesis about this being an eye disease as opposed to a brain disease, you can't fault people for thinking this way, because obviously the first thing you would think of when you think blind is, oh, the eye is bad, right? It's a very easy connection to make, a very easy hypothesis to identify, right? And so it's a good story to say that Hubel and Wiesel's contribution meant a lot in order to develop appropriate treatments for people with this condition. And that would be a good story. However, history is complicated, and I want to point this out. Digging into the literature, I found that doctors were experimenting with occlusion therapy as early as 1920, much earlier than Hubel and Wiesel, right? There were doctors in the 1920s who had no idea how the eyes were wired to the brain. I mean, some idea, right? They knew anatomy and stuff, but in terms of like how vision is interpreted in the brain, they didn't know how that worked. They knew that the eye was wired to the brain, that's about it. And even then, in the 1920s, there were doctors who were experimenting with giving patients these eye patches and seeing whether or not they improve their vision, much earlier than Hubel and Wiesel, right? This is 30 years before those first cat experiments. So occlusion therapy involves covering the strong eye and allowing the weakened eye to strengthen, right? It's a pretty simple approach, but it was also kind of inconsistent back then. So while these Nobel winners provided the mechanistic insight to double down on occlusion therapy, medicine is its own field of research, even if you can't find a mechanistic explanation. Now, does that mean that the work of Hubel and Wiesel did not contribute to developing treatments? No, right? This is a nail in the coffin that convinced everybody that this disease was not an eye disease, it's a brain disease. But like, medicine and science happen at the same time, and medicine got to the treatment before science did. Science provided the rigor, science provided the justification to invest more resources in developing this kind of treatment. So I want to shout out the doctor scientists of old. Like, there are a lot of wacky stories from the field of medicine, right? A lot of doctor scientists in the past were just flying by the seat of their pants, right? Like, hey, who remembers bloodletting, right? They were just trying stuff. But the thing is that the records of the doctors that were experimenting with occlusion therapy, they were recording everything they could. Like, I think a lot of doctor scientists, even if they didn't end up being correct at the end of the day, were excellent scientists. They were just working on different problems than traditional research scientists, right? They were recording everything about eye responses, right? They were doing therapies and they were recording things like pupil dilation, the time it took for the pupil to dilate, the time it took for focus to break, the time it took for the eye to twitch. They were measuring everything because they were flying blind, because science was lagging. They just had to go with what they had, right? And this is like the indomitable human spirit, right? The indomitable human spirit. The drive to improve things, despite it being so difficult without mechanistic understanding. Again, these doctors that were giving kids eye patches without understanding why they were giving eye patches, based off of a hunch, based off of a guess on how the brain worked, they were being very, very diligent, keeping records and measuring, because they were driven by a motivation to fix this issue for these patients, right? That is dedication, right? They might not have known completely what they were doing, but they were ferociously dedicated to helping these patients. And I respect that, right? I respect the hell out of that. So even if the doctors in the 1920s didn't know how the eye and brain were connected, they still cracked out therapies 30 to 40 years before a mechanism was discovered. It's another example of how medicine and research, medicine does not always rely on research. Research does not always rely on medicine. They do help each other, but they are individual disciplines, right? They work differently. Medicine is different, right? The purpose of the field of medical research is to help patients. If they find something cool about the brain, that's almost an accident, right? It's not to understand biology. The purpose of medicine research is to find a gosh dang cure. It's to make a real difference in a real person's life. And you don't always have to know what is happening under the hood to make that happen. And as you can see, like, they didn't know how things were happening, but they were like, okay, we just record everything we possibly can. And if certain metrics get better, we know we're sniffing around in the right direction, right? They were flying blind with a flashlight, but they were using the hell out of that flashlight. So it amazes me that they were able to come up with occlusion therapy 30 years before they knew they were right. Crazy. So what have we learned since then? Obviously, a whole lot more about how the brain is organized, mechanisms of plasticity. So if you've taken any amount of neuroscience, you know these terms: long-term potentiation, flash depression. These are ways that neurons tune their firing strength to each other. The developmental process by which the visual system is refined. Like we already talked a little bit about how your brain is not developing pretty much ever, right? But when you are born, there is a large period by which you have the capacity to change the connections in your brain. There's a lot of refinement going on, right? A lot of refinement. So the process by which your visual system and the brain as a whole is refined has a lot to do with this cat research. You don't need to know how a car works to drive a car. Reminds me of that saying. Exactly. Synaptic pruning. This is the process by which extra connections on neurons are cleaved, or cleaved is kind of severe, maybe trimmed. And, it wouldn't be my channel if I didn't mention microglia, right? I love the fact that the immune system is doing double duty in the brain, right? The immune system can't do a lot of its crime-fighting stuff in the brain, because if it did, it would be really bad for you. It would cause your brain to get inflamed and you'd have damage and stuff. That's not good. But there are specialized immune cells that are local to the brain called microglia, and they do a lot in the brain beyond crime fighting. They're also involved in making sure that your brain develops how it is. They chew off extra connections between neurons that are not needed. And that is really, really important, because if you have too many synapses, that's also bad, right? In order for normal development to happen, you start off as a baby with a bunch of these synaptic connections, and then as you develop, you trim them, right? I think there's this common misconception in like Hollywood that like, oh, this genius has like, double the number of synaptic connections. They just have so many synapses compared to the average person. They're a huge genius, big brain. No, you want the correct number of synapses. Not too much, not too little, the correct number. Okay. That's it for my presentation, and I will end with these two shout outs here. So we have a 12-hour charity stream in January. It's probably going to be the 10th of January. Well, I will be streaming from 9 a.m. to 9 p.m. EST, covering, I don't really know what to call this, but a collection of diseases of female biology. So breast cancer, ovarian cancer, and endometriosis. This is both an awareness kind of thing, but mostly it is to dive into the research and to help raise money for research in these causes, right? I think that, speaking like behind the scenes, right, in terms of why do I pick certain topics, I pick certain topics because I find them interesting first, important second, right? I could not force myself to cover a topic purely out of like motivation to do good. These conditions, every time I learn about them, my mind is like blown. So if you are available that day and want to pop in and learn about these conditions that affect a lot of people to a very deep degree, but also if you are intrigued by the manner in which these diseases function, come hang out with me on that big day. Big day. Yeah, I don't really know what to call this. Female reproductive health, female health, I don't know. And then another shout out happening tomorrow at 1 p.m. EST. There is a collab stream between Z and Sidray, which I think is very, very fun. The synaptic pruning, it reminds me how in certain languages don't have certain sounds. We can all make these noises with our mouths and vocal cords, but if you don't use them eventually, it's weird. Yeah, that's just like plasticity and learning, right? Like if you use it or you lose it, right? That kind of thing where we are constantly trying to make efficient use of our brain. Efficient use. Gynecological health, I don't actually know if, like, this is just pure ignorance. I don't know if breast cancer counts as gynecology, but maybe it does. Yeah. The other day I learned you don't even need a uterus to have endometriosis. It is fascinating. Like endometriosis is really, really interesting. It's also devastating, right? It's I don't remember the statistic, but it is responsible for a pretty gnarly chunk of infertility issues. And even if you were never planning on having a child, it hurts like beyond belief, right? People with endometriosis have what can be debilitating pain. And I think that sucks. And I think the process by which we quote unquote solve these conditions, like it all feeds into each other. These are conditions that all have to do with cell fate. And I don't know if I'm establishing a kind of pattern across different charity streams, but I find that I'm more amenable to covering cancers and diseases of cell fate on stream as a charity than in a video, just because again I don't like feel super comfortable joking in a cancer video. Yeah, all right. Let me get your reactions to today's stream. What did you think? Did you learn cool things? Is there anything that you think will stick with you? Learning about cats and how they contributed to our understanding of the visual system. Any particular standout topics or anything that you would like elaborated? I'll try my best. Or if you can find something that I can't elaborate on, that's a W for you, right? I used to think that way as an undergraduate, where I didn't like raising my hand in class because I didn't want to take time, right? The professor has a lecture to get through, so I didn't like holding up the lecture just to have a question answered. But I did try to ask the professor one question at the end of lecture, just as a way to like challenge myself. And if the professor didn't have an answer, that was like a little hit of dopamine for me, really. It was like, I found the limit of your knowledge, which is not a bad thing, right? It's fun to like see where my stuff ends. I love my unrelated degree, but these videos always make me wish I studied biology. I mean, that's what I'm here for, right? I can help you study biology without you having to do the miserable part of learning every step of the cell cycle, learning every metabolic intermediate in glycolysis, right? My whole thing is I want to provide you with the fun parts of biology. I learned that vision has layers. Yeah, it is super cool. Like there are good resources online that'll show you how vision progresses through the layers to try to give you an idea of how your brain puts together an image. It's really, really neat. I think it's kind of cool. But I thought we were getting a presentation on cat biology. It is a little misleading, but my community did vote on this particular topic. It was either going to be the way that cats have informed us on how our eyes are connected to our brains, or it was going to be on the genetics of cat fur coats, the coloring on their coats. Because the way that cat color is inherited is super messy. As if you've ever known a cat, a stray cat, you can see that they often have litters that look nothing like either parent in terms of coloration, and that is it was my excuse to teach about genetics, but I'm kind of glad that we did this instead, because again, my research was in synaptic pruning and synaptic plasticity. So I was a lot more prepared to do this than remember how cat dominance, recessiveness, and all that nonsense in cat genetics goes, because it is really, really stupidly complicated. Yeah. I missed most of the stream but got the conclusion, so I'm happy. That's cool. Yeah, I mean the story is quite good, but the idea of like cats winning such a significant Nobel Prize is, I think, amazing, right? And again, like if you're worried about cat health and their usage as a research animal, we don't really need to use them quite as much as often anymore. Mice can do that job too, or rats can do that job a lot of the time now that our tools are better too. Wasn't there another research where you gave kittens striped glasses, and if you turned them 90 degrees, they become blind? I did read about that, but I'm not prepared to talk about that because I needed to read that. I really prefer, as much as possible, to read the primary sources for this kind of thing. And I did not, I just couldn't, not with that one. Vision having algorithms to simplify the problem is pretty cool. I wonder now if we've incorporated the algorithms in computers now. I believe we are. I think edge detection is like the first layer of machine vision. I don't know how similar machine vision is in its entirety, but I know it is similarly schematic. I'm more curious about what is going on physiologically during the critical period of neuron development and what changes to trigger the cutoff of that period. A topic for another day. I had the answer at some point in graduate school, but I'm not nearly as confident in that knowledge nowadays. But yeah, I think that would be a good topic for a journal club. I like doing neuroscience in journal clubs too, because I think it's really tough to do it for a video, because it is so, look at the brain, look at this region. Not quite as fun and poppy, I think. I didn't think plasticity was that big of a deal in newborns. Do you think it works similarly with smell or other senses? I wouldn't be surprised, because there's a lot of plasticity that happens in newborns, right? Babies are learning machines. They are learning machines, right? You can even see evidence of this in language acquisition, where babies are like responding to different languages in different ways. Like they are goated. They can learn. They are incorporating a lot of information all the time. Yeah. This was a really fascinating stream. Thanks for coming and hanging out. Are you now a Phyllis Caddis neutrophil now? That I am. I would love a video about cat color genetics if you want to do it sometime. I might do it as a charity incentive for the next 12-hour charity stream. Don't forget, cats can have multiple fathers in the same litter, much like dogs. I didn't know that. That's pretty cool. Cat genetics are that hard? If you thought like dominance and recessive was like fine, I mean, that is fine, dominant, recessive, that's cool. But then you get into this idea called epistasis. Okay, and then we have to talk about which gene comes first in the epistasis pipeline, which I think is actually really fun, but it is also really complicated, right? Some genes mask other genes, some genes require other genes to work. Like there's a whole, and again, epistasis is really important for understanding how genetics works. So it's a really good time to dig into that. It's just the diagrams the diagrams of how you get a calico cat or whatever. It's like, it's crazy. Yeah. I just went over cat coat genetics in my course. That's awesome. I actually never learned it in a formal setting. I mostly picked it up on my own. But yeah, it's not straightforward. It's not as easy as tall pea plant, short pea plant, like Mendel did it. Did I hear about the new coat variant? I did not. Could do a journal review of that striped glasses thing. Yeah, I think I could. I think that would be cool. Will I ever cover how bees have diploid gene systems? I don't really know enough about bees to figure out a cool angle on that. Hi, is this the cat fi that the VTubers have all been talking about? Hey, Charles. What's up? What's going on? You've caught me at the end of stream, having finished talking about cats. The cat fi. Yeah, the cat fi has made an appearance. A rare appearance, by the way. I reserve this only for when certain incentives have been met. So here I am. Fi the Neanderthal. So developmental disorders are a problem with plasticity. Not all of them, right? I mean, it depends on what you are talking about, right? Plasticity can be an aspect of certain developmental disorders for sure. Are you talking about Toxoplasma gondii? Nope. We talked, I guess it's past tense, talked about how cats have contributed to our understanding of the visual system. Toxoplasma, I will do as a full video at some point. Trust me, I will. Just not right now. Will I do a video on a parasite like Plasmodium? Yeah, Plasmodium is definitely on the list as well. Oh man, like I've been saying this on stream multiple times, but there are more interesting biology topics than I have a lifespan, right? I will die someday, or rather neutrophils die every day. So rather my lineage of these neutrophils will die one day, and I don't think that even after a whole lifetime of content, I could work my way through all the interesting things in biology that I want to talk about. Yeah, so don't worry, Plasmodium will be covered. What a headache though, because okay, let me rant a little bit about Plasmodium. Anytime you talk about a parasite that has multiple hosts, you have to do twice as much work. Oh, you have to do twice as much work because not only, so Plasmodium, for those of you who don't know, that's malaria, right? So, you know, malaria has part of their life cycle in the mosquito and part of their life cycle in the human. So I've been putting off malaria because it's just so freaking complicated. And I'm not ready to do a video that complicated quite yet. It wasn't a striped glasses, but an entire striped environment. Yeah, I did remember reading about them. You can read about them in the blog post, I believe, that I linked in the description of this video. Yeah. Oh, okay. Let's get big. Let's hide that PowerPoint. Transition into just chatting. I miss just chatting. I feel like I really enjoy doing these like information streams or learning streams, but I also just like yapping for a little bit. And I think because the last couple weeks have been so stream light, I'm like, oh, I miss just yapping, just catting. True, just catting. We are deeper into the school year, about to head to the Thanksgiving break for those of us in America at least. So I'm wondering how your school classes have been going. Any cool things that you've learned? Any cool classes you are taking? Why do neutrophils still attempt to fight off viruses even though they are not effective at killing them? So I say this quite often in my videos, but biology is just weird shapes bumping into one another. So if a virus has been opsonized by antibodies, right? So, viruses float around in your system, your B cells make antibodies against them. And if a phagocyte like a neutrophil sees or doesn't see, it doesn't have eyes, but if it detects an antibody, it'll just eat whatever is inside the antibody, right? It's not consciously trying to fight viruses, but it is fighting whatever the B cells are directing them to fight. Another good lecture today. Thanks. Oh, I think I forgot to say hi to you, Penbu. Welcome, Penbu. Penbu and I did a collab very recently on Penbu's channel. So, oh gosh, sorry, I didn't shout you out earlier. But this is partly incentives for the marathon stream that's happening in January, where one of the incentives is going to be an original song that I will try and write mostly on my own. I think it's fun to like try to live up to this idea of like a science VTuber idol, and Penbu was really instrumental in giving me my first lesson in composition, because Penbu does a lot of music composition and sheet music stuff. So if you're interested in music and how music is constructed, go ahead and give Penbu VT a plug. Let me go ahead and plug your channel real quick, because we did that collab recently. I like don't have a very rigorous background in music, so it was really informative for and really helpful because like, could you teach yourself music? I mean, I guess you could teach yourself music if you're crazy. But having a teacher is way better. Our friend Michelle just did a talk in Toronto about her research with mosquitoes and West Nile virus. Oh, that actually overlaps a bit on the stuff that I was doing in graduate school, where West Nile virus has a brain component to it too, along with Zika virus, right? These are bad viruses to get if you are expecting. Famously, Zika, right? That was a nightmare. Um, so that's interesting. That's cool. That research is very, very important and also very much in my wheelhouse. It's a virus, a virus I will eventually get to one day. Very, very neat. Toxoplasma is closely related to Plasmodium because they're both protozoa. Or are they like not close at all? I mean, there's a lot of diversity in protozoa, right? They are a category of life that I'm the least familiar with, so I hesitate to draw too many conclusions. I'll put it that way. He did very well that stream. Thank you. Expect good things. Thank you. Thank you. Yeah, I've always wanted to make my own music, but having played with music in high school, the stuff that I composed was just garbage. And Pembu gave me a lot of good tips as to how to break out of this problem I had where like I would write a chorus, but then I didn't know how to write a verse, right? I think we've all had moments where we've had ditties come to us, and you know, a ditty, a little short tune might not be that hard to think of, but like going from okay, this is one part of the song to this is another part of the song, I'm just writing more chorus. I don't know how to make a different part of a song. My brain was like breaking, and Pembu gave me excellent advice as to how to get past that block. And so I'm excited to show you what I can do, but only if we reach those incentives in January. I'm doing a course on mammals, and I'm supposed to do surgery on a shark next week, but I have big hands and poor eye coordination. I do feel for you. I have bad dexterity myself, but even I was able to do like brain dissections in mice for my graduate research. You'll be able to do it. You just have to come up with some hacks, right? If I were a hand cam streamer, I could show you the way that I stabilize my hands. And it often relies on pressing your hand against something or your forearm, something to reduce the degrees of freedom that your hands can tremor. So, you know, there's my tip is find a way to stabilize yourself. Like play around with the orientation of your forearms and wrists and stuff to be able to do the dissection. What I would not recommend is I would recommend having your elbows somewhere solid to prevent, you know, an incredible amount of movement from happening. You know, obviously if you're not going to be a neurosurgeon, you don't have to have free stabilization of your arms, but if you're just doing dissections, keeping your holding sticks stabilized somehow is really helpful. If being a composer would be peak. I came up with a melody for that Blender song in my Black Death video. If you haven't seen it before, here's the plug. I did a video on Yersinia pestis. People have been asking for the Black Plague forever, and I did it. And in that video, there is a music segment that I wrote the melody for, I wrote the lyrics for, and I came up with the general vibe. But I didn't like write the bass line. I didn't, you know, write the piano part. I didn't do any of that. I collaborated with some very talented musicians, Keko Gecko and Rain Napper, to actually make the track. And the difference now is I want to do more of it on my own because I think it's fun. And I think it would be fun and more meaningful for my audience to hear music that I myself have made, with the goal of it being completely from myself. Because if you've watched my members video, I did a members video on the definition of art and craft. And I think it's just more meaningful if everything is done by as few people as possible, for the authenticity of the art itself. When you watch my video, you are watching a reflection of me. This is what I can do with these tools. It is pure undistilled me and pure undistilled Toxo, because Toxo draws the original assets that I use. Right, and I think it's important that for a piece of music that I eventually be the one to see that project from the beginning to the end. So I'm excited to show you what I can do too. Protozoans aren't even monophyletic. That's also interesting. I didn't know that. I know so little about protozoans. If neutrophils didn't attempt to fight off any viruses, then mortality rate from virus infections would drop significantly because inflammation wouldn't happen very often, right? I mean, not always the case, right? Neutrophils do other things too. It's kind of hard to make a hard conclusion like that right off the bat, right? Neutrophils, they have a primary job, which is to fight viruses. Not fight pathogens, I should say. But in something like a viral infection, I would say oftentimes they are doing more harm than good. From my limited reading, right? I'm not an actual immunologist. I'm like a wannabe immunologist. So take it with a grain of salt. But they're also involved in like wound healing and stuff, right? So you can't have the neutrophil be totally uninvolved in the process. And they're also frontline detectors of infection as well, right? They, not to the extent that macrophages are, but they do play a role in helping the immune system get up to speed, right? A neutrophil detects some bad protein that they don't like. They will spit out inflammatory responses to trigger other responses, right? So it's not as clean as, oh, if only neutrophils just didn't attack viruses, then everything would be cool. It's more like neutrophils play a multifaceted role. Every immune cell plays a multifaceted role in the immune defense. And like, you can't really, it's really hard to just prevent them from doing one thing over the other, right? But in terms of like, I can see the vision of like, if you had a drug, for example, that calmed the neutrophils down, didn't kill them or anything, but calmed them down, then that would work. And I think that is what you are getting at, right? I think that is what you are getting at, Fale. Diddy, sorry, that's just a word for like a little jingle. I should probably say jingle because the word Diddy is poison now. The Protista kingdom is just a bunch of single-cell eukaryotes. Biologists didn't know where to fit in. That makes sense to me. I think they do feel like a random grab bag. Back, been gone for like 45 minutes. Well, we are just hanging out and chatting right now. I would also say that because I don't know every viral infection, I can't make a conclusion based on every viral infection. It's just the few viruses that I have read about, neutrophils have not exactly been overall that helpful. But again, fighting a virus is like multifaceted, right? There's fighting the virus on the front line and then sending out an inflammation signal so that the rest of the immune system can come and fight. And then there's prolonged fighting where the neutrophil just keeps on reacting angrily, and then that can cause problems. So I think the story is complicated. This is how we do it in photography too. The more points of contact, the more stable your shot is going to be. That makes a lot of sense, right? Like anchor your elbows, make sure that they don't move. I do soldering and disassembling electronics. I do a lot of bracing my hands or arms to reduce shaking to get better. That's really cool that we all converged on the same technique, right? Like to get fine motor control, you have to limit the amount of wiggle your arms can do. Life was better before Blender. Yeah, I really had a lot of fun making that song. Isn't a little jingle a ditty rather than a ditty? In American English, I think double T and double D sound very, very similar. Like even single T and single D, right? You know, there's water. You can say it water. I think that's more like European, but in American English is water, water. Kind of a little weird language specificity. Water, yeah. My water. I don't know why American English is like that either. But yeah, did anybody who listened to the Blender song, were you able to tell what song I based it off of? This is kind of an interesting poll. Do you have any like, because I was so caught up in being like, oh, people are gonna think I ripped this song off, this like very famous song, and they're gonna think I parodied it, when in reality I only used it for inspiration. So I'm wondering, when you listen to the Blender song, did it like trigger immediately what I was going for? And you don't have to hide, like, you don't have to say the answer, that would make me happy. But like, was there a song that popped into your head that the Blender song was reminiscent of? I was trying very hard not to have it be a direct ripoff of the thing, but I'm not really a good judge. Okay, no clue. Okay, nice. Nice. The song sounded familiar, but I can't tell why. Does anyone, okay, maybe not song, does anyone have an artist that they think I modeled that song after? an artist I took inspiration from. That might be a little easier. Might be a little easier. It might not be. But if you listen to similar music, you might be able to tell that there is one particular artist who did one particular song that I really, really liked, and then I just like totally ripped it off. But this is kind of going to a conversation that Penbu and I had on stream where like nobody is truly original, so I guess I'm not surprised that people can't tell. But it does make me feel good about like, okay, the fact that you don't know which song I was inspired by means I added enough of my own creativity to mask it so it's not a straight, rip off, which is nice. It is nice. The style, there's a massive copyright discussion to be had too. Yeah, that's true. Maybe it's obvious and we're just bad at remembering. That's also true. That's another possible explanation. But yeah, I didn't sample the original song or anything. It was my own original, like, melody, right? I think the melody was mine, but the chord progression, you can't copyright I mean, people have tried to copyright a chord progression, but the chord progression I got from a song by Joji. It's like, this like lo-fi kind of sad boy kind of aesthetic. I really enjoy that aspect of Joji's music. But the lyrics are completely my own too, right? Like I want to make original music, so I didn't, because I thought, oh, I could just parody a song and then write lyrics over a song that exists. But I was like, it's more fun, I think, to come up with something original. Slow Dancing in the Dark, yeah, exactly, stuff like that. It wasn't Slow Dancing in the Dark, and I guess it's technically not correct to say Joji. Technically, this song is from Pink Guy, Joji's former personality. I don't remember what the song is called, but it's the one where it was like, I live in a constant state of fear and misery. That one. Whichever one that song was. I love that song. And I think if you play that song and you listen to my song, you can kind of tell like it is quite similar, but not enough where it would evoke like, oh, this is just a ripoff of that song, which is nice, I think. Yeah. Yeah, I find the process of making these little tunes really fun too. Like it is, and I was going to do like this next one. I'm aiming to be a full song because the Blender song was just me practicing, right? Just getting a little practice done. Just like when I started this channel, I did YouTube shorts to practice video editing. I eventually do want to have that original song, 2 to 3 minutes, something catchy and fun and silly, and the purpose of the songs, the purpose of the whole channel is just to make content that makes people smile at biology. And I think this song that I've planned is very hopefully in that vein. You can even hear Pimbu transcribed it at the end of his stream. You can even hear a sneak peek if you want to peek over at his channel and get an idea of what I was going for. Yeah. I hate it when pathogens are human specific and can infect a lot of people, but most infections are asymptomatic, yet it chooses a small subset of people to suffer its symptoms. H. pylori did me dirty. Yeah. Yeah, like human only pathogens are really hard because, you can't really study them very well in other species. Like gonorrhea comes to mind as a human only pathogen. Sure, you can infect a mouse with gonorrhea, but they're not their natural like prey, right? So it's a little tricky to do that kind of research. And it's, that's part of why Plasmodium is so hard to study, right? Malaria, it survives in mosquitoes and it survives in humans. And if you don't have it in either context, who knows how much your conclusions that you draw in a dish can be applied to actual real biology. It is really, really difficult to do malaria research. You said pink eye, my brain floated towards STFU. Yeah. core memory triggered. TBH, modern cladistics just kind of abandons the concept of fixed ranks, we just keep them for convenience. Dude, phylogeny just like breaks my brain. There's a lot of phylogeny discussion in this upcoming video of mine about giant viruses, which I am teasing. I hope it's okay. Let me message because you're here. How do I DM you? Where do Yeah, so if you want a sneak peek of what I'm working on nowadays, it's a video on giant viruses. And like 4 years ago, everybody, all these biology channels were talking about giant viruses, being like, oh, are they the fourth domain of life? Do they blur the line between living and non-living? And it's been a while since anybody that I've seen has talked about giant viruses. So I figured it's good to give you all an update as to what is going on in the giant virus world. Why do you do a lot of music production, or was this a one-off? I, like every sad boy in high school, wrote a couple of songs, specifically just the chorus of those songs, without much of a music background. So it would be unfair to say that I have zero music experience, but I have basically zero music experience. And so music production has always been like a bucket list thing for me to do, because I find it very appealing to make a song that comes out of my own brain, but I've never had the chance/reason to do it, and now I do. The Blender song was the first song that I showed anybody, so that was like a big major step. Mimi virus will make an appearance in the giant virus video. That's how you get the whole debate on are mammals fish. It's just so everything I've said on this channel, right? Biology is a spectrum. Words are words. They're just ape noises that somehow, whatever. Okay, I've been given the okay by Charis Cars Euphron. I'm just gonna call you Euphronor because I don't remember how to say your first name. My buddy in chat here, who I've collaborated with on his channel, a philosophy VTuber, which I love. I love that there's a philosophy VTuber out there. And the thing about giant viruses and also subviral infectious particles, which is the next video after giant viruses, is that the idea of a virus breaks so many things in terms of our conception of categories that giant viruses, yeah, they challenge our definition of what it means to be living or non-living. And then subviral infectious particles really challenge our idea of what it means to be living or non-living. Things that are smaller than viruses, that are virus-like, are so interesting and so cool. So keep an eye out for that. And I'm teasing a collab. Don't know when it's happening yet, but I do want to sit down with our resident philosophy VTuber, Karis. Okay, Karis. Words are made up anyway. Thank you. That is the thesis of my channel. Words are made up anyway. I mispronounced archaea in my archaea video. I called it archaea in my archaea video. People called me out for it being archaea. I looked it up. A lot of people say archaea. More people say archaea than archaea. In America, it's prion. In the UK, it's prion. Pronunciations are made up, but I will try and say your name correctly. Karis. Our boy Karis. I am planning to have him on this channel to talk about the definition of life, because as a biologist, I have a certain perspective as to what counts as living. Not to say that I speak for all biologists, but I certainly have spent a pretty good amount of time thinking about the construct of life. Subviral infectious particles, would prions fall into that category? It would, but I'm not focusing on prions because I already made a whole big video about it. Although maybe I could put prions in the thumbnail and people will click on it again. Is that too... damn, that's a really good idea. I've already had a prion video, but it's been a while since that prion video came out. Maybe I do put prions in the thumbnail and talk a little bit about them and then redirect them to that prion video, because the prion video that I made, I think is really good. And I think it still holds up. And if that went viral again on the algorithm, I wouldn't mind. So yeah, prions do count as subviral infectious particles, but there are more than just infectious proteins. There are, I don't want to spoil. I'm not going to spoil. There are biological entities smaller than a virus that are infectious, that can cause disease. And I can't wait to tell you about them. In fact, like me doing a giant virus video, I knew I had to do these one after another because the way that they segue into one another is really, really cool. Yeah. I say prion in the US, AKY. It could just be whoever taught you initially, right? Like so much of our language is passed down. And the other thing is I recently recorded a thing where I had realized that there are words in biology I have never seen, that I have read, but have never heard out loud, right? These are textbook words, words that are used in textbook settings that are not typically used in lectures as things that you hear. And so I'm just like, oh, I say these things because these are just the ways that I've heard other people say them. It is cultural dissemination, I believe. Mention prions, they're well known. Yeah, yeah. I think so. I think it's a good idea. A lot of words I've only ever read, so IDK how to pronounce them. Yeah, exactly. Just English is such a nonsense language, especially English, right? It's a mishmash of multiple different heritages, borrowing so many loan words and converting them into our own lexicon without any consistent pronunciation or rules. It's like learning several different languages at once. So, yeah, I'm not going to get mad at anybody for pronouncing certain things incorrectly in English. And if people get mad at me for pronouncing things wrong, I don't really care. And would you believe it, there are still people picking on my pronunciation for these things in those older videos as early as like a week ago. Based opinion. Yeah, words are just ape noises. They're just ape noises that the majority have agreed are the correct ape noises to make for certain ideas that are infinitely complex, and so you can never capture all of their nuance just using ape noises, but they are useful. They are pretty good and pretty useful. Yes, if enough say it away, it is a way. So long as it is mutually intelligible, it doesn't super matter. Exactly. As long as the idea of what I am trying to convey in your brain is conveyed and conveyed in about the same amount of processing time, it's fine, because there is a limit, right? There's a limit where if you, I don't know, say it like I don't even know how you can Orchior or whatever, Archaea, like that, you get what I mean very quickly. If you really butcher it, you can interfere with processing time, and that can be kind of annoying. Phonetic languages make it easier, I guess. Easier for sure. But even then, it's not exactly standardized, right? Like you have like you know, a standardized set of syllables in Mandarin, for example. And you know, if you've ever tried to learn Mandarin, there's a very standard sounding, oh, if you see this tone, it makes this sound, and then you think that, okay, so that puts a constraint as to how Mandarin sounds. And if you go to China, you realize that you can put weird variations on tone and articulation, even in a language where it is supposedly standardized for sound. I wonder if there's an evident critical mass of people that are needed for that to work. It's like, I've been thinking about this idea of cultural right makes right, right? Like, as long as you have cultural dominance, it's like you can, not to say that it is good, right? But the way that power works is power lies in the majority. Even if the masses don't believe so, power still lies in the majority of people. So yeah, there's probably some critical number out there. I wouldn't say there's one right way, but there are wrong ways. Yeah, I think that's a pretty good simplification. That's a pretty good explanation for that. Portuguese has so many sounds for the letter X. Oh, I can't even imagine. Oh God, Karis, why? If enough say it away, it is away. So long as it is mutually intelligible, it doesn't super matter. I'm you okay. Next time you stream, you have to install a mod pack for whatever game you're playing to display all the language like that, and and you have to just be happy with that. You just have to eat that mod pack next time you go on. I hate that I understood that. It does weird things to your brain. Male fashion is full of examples where an individual did a thing and now it's frowned upon to do it the other way. Yeah, that is true. I will say that. It took me a while, I think, to get used to Gen Z fashion. And now I dig it. I get it. The big pants, I get it. I didn't get it before. My millennial skinny jean brain didn't get it for a while, but now I get it. The long pants, the big pants, that can stay. That's actually such a good idea. Yeah, you know, I got ideas, man. Oh, okay. You have to play a dating sim with a language mod pack that ruins it and try to get the good ending. Jinko has entered the chat. I wonder a lot about this particular topic, right? I'll talk about this one last thing and then maybe call it a stream. This is an idea that I've had in my head for a very long time, and my thoughts about this topic have evolved over time as well. And it's this idea of cultural touchstones, this idea of all-time greats. And the reason why I'm thinking of this is because this idea of cultural fashion was brought up. Because, dude, when I look at old episode of Friends, man, like the 70s and 80s, people look back at that fashion and go, wow, that was a mood, that's a whole aesthetic, that looks great, bring it back. I look at the fashion from Friends, like in the 90s, millennial coded fashion, and I'm like, oh man, dude, we were not cool. That hurts, man. Because even though they might not be themselves millennial, I don't remember how old they were back then, the friends of friends. That was the fashion that we grew up looking at, like these, God, unflattering. 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 I miss looking cute after transitioning. My new clothes are so bland. Is this about male fashion? Yeah, male fashion, I feel like there are fewer degrees of freedom, but I do respect Gen Z for trying a bunch of stuff. Um, Not just Gen Z fashion, but also leave the last button open on your suit. Or male colors are black, beige, navy, and olive. Oh, yeah. I mean, the thing is that if enough people believe something, it then becomes our perceived reality, right? Because it doesn't make it less real to think or to feel, I should say feel, to feel that the last button on the suit doesn't look good. It still creates a reality that your mind perceives. So, yeah. Why put the extra button on it? Yeah, it drives me. It used to drive me crazy as well, but I kind of get it over cultural indoctrination. But yeah, this idea of cultural relevance, I've been thinking a lot. I want your guys' take on this because maybe this becomes a members video at some point because I do find myself ruminating on this idea. Like fashion has always been cyclical, and I think that that's fine because I think fashion will always be cyclical, and I think fashion is an art form where that might be unique because I think every time a new generation is born, they're going to get fatigued on whatever their parents were wearing, right? Or whatever their grandparents were wearing. That's just not going to be cool to them because they want to experiment and self-express, right? You don't want to put on the same clothes as your mom or your dad or your grandpa or grandma because clothing is a method of self-expression and you are not your parents, right? I don't think it can be simply explained by an expression of novelty like, oh, I don't want to wear these clothing that look good because I just want to be different. I just think that you associate those clothing with an older generation and you do not identify with that older generation, so you need to carve for yourself a new way to express yourself. I think that's valid. I think that makes sense as to why fashion goes through cycles because there are only so many permutations of shapes that have some kind of visual appeal at some basic level. But I think the reason why things come in and out and have these reoccurring periods is because there is a kind of fatigue, generational fatigue, I think. I think where I am interested in, and you can buy that premise, you might not buy that premise, but go with me here, stay with me here, right? What about music? Right? Are there musical generations left? And I find myself wondering this because, right, if we look at the 50s, if we look at the 60s and 70s and 80s, there's a vibe, there's a trend, there's a hot new genre that's taken the nation by storm. And we get to the 21st century, and in our unprecedented ability to both be hyper-connected in the form of the internet, but also hyper-divided in our ability to form subcultures, have we reached a point in music development where we no longer see these generations of music? Rather, are we seeing a tribal splintering of music? Like maybe there's no more generations of music, but just a neighborhood of music. Does that kind of make sense? The reason why I say this is I really wonder what millennial music will be remembered for, right? Because if we look back in the past, we can say Elvis, right? One of the all-time greats, the Beatles, all-time greats, Beethoven, all-time great. Who's the millennial all-time great? Right? who's the millennial all-time great? Who will we look towards as the music of our generation? Because I think for most people growing up in America, you could yeah, was Elvis Presley one of the greats? Yes, they would probably agree. Even if they didn't like Elvis's music, they would probably agree that Elvis had a significant impact on the music scene in America. But like 90s pop? Like 90s had grunge, right? Grunge music, that's a generational genre, I think. And then we get into this pop era where, yeah, there are new genres, like mumble rap was a thing, right? But does mumble rap define a generation of music? I don't know, right? This new label of pop, right? I guess 90s and early 2000s, there's boy band music. I think that's fine. I think if we are remembered for the introduction of a boy band as a genre, but is boy band even a genre, right? That's kind of interesting too, right? Is that even a genre? Is that even a genre, right? Because we have K-pop, and a lot of K-pop is, you know, reminiscent of boy bands. Is K-pop distinct from boy band as a genre? Yeah, you know, I don't know the answers to these questions, right? I don't know. But what I do think is interesting is I don't think we have the cultural consensus left to call any artist, musical artist of our day, the voice of a generation or the sound of a generation. That's my take. That's my take. I don't know if it's a good thing or a bad thing, but I don't foresee Justin Bieber being remembered as fondly as Elvis Presley, right? Like, you know, who's our chat? Who's our Elvis Presley? Who's our Elvis Presley? I think it's an interesting conversation to have. You no longer have a significant portion of the population listening to the same stuff. Yeah, because we have the internet now. And even though we're more connected than ever before, we're also more splintered than ever before because we can choose where we go. Music today is fragmented in several niches that have nothing to do with what plays on the radio. Yeah. What's millennial again? I think born after or born before, what's the cutoff? Like 1996-ish? For, you know, a generation worth of time, that's your cutoff. Rough cutoff. Okay, and words are just ape noises, so a millennial is whatever a millennial vibe is. Sorry. Hyper-specialization algorithms leading to microculture. Yeah, exactly. Like, I don't know, has that occurred to anybody else? The fact that, like, we are not gonna really have a Elvis that people are gonna be happy to call Elvis, right? Like, you could make an argument that maybe the last one was like Michael Jackson. I think that you can make an argument for that. Michael Jackson, voice of a generation, widely loved, right? Omega star. After Michael Jackson, I don't know. I'm not like a music pro or anything, but I suspect that, you know, if you say it's Taylor Swift, a lot of people are going to say no. If you say it's Justin Bieber, a lot of people are going to say no, right? If you say it's Nickelback, a lot of people are going to say no, right? And I think that's just kind of interesting. I think that's interesting. Now the nostalgic music is punk pop from the early 2000s. I have heard that. I don't know enough people in Gen Z to witness that for myself, but, you know, it is interesting. Hip hop has shifted away from being defined by the gatekeepers, replaced by public figures of the youth like streamers. That's interesting. Yeah, hip hop has its own unique culture as well. Still very innovative and not as maybe corporate as, you know, the genres that I'm thinking of. So that's gonna be an interesting take. But because I mean, maybe this is a hot take, but I think hip hop is a little more underground than like Elvis was comparatively, right? I'm talking like big, you know, worldwide, or at least countrywide phenomenon. And hip hop, I'm not discounting its contribution to the music scene, but will we collectively as a generation agree that hip hop is our generational music? I just think, not to say that it isn't good or worthy of it, but could we get enough consensus that it is? Can an entire country be a genre? I think it's interesting to debate whether or not K-pop has enough signifiers to warrant its own unique categorization. I also don't want to be destroyed by K-pop stans, so I'll just leave it as an interesting idea. Like, please don't make me say Taylor Swift. I know, right? It's almost there's almost a reluctance to say that the millennial Elvis is Taylor Swift. I'm like, there's gotta be somebody else. There's gotta be. There just has to be, man. Um, and that struggle is why I find it so interesting, right? Because I don't think older people would struggle to say it's Elvis. I don't think they would struggle to say that. Yeah, yeah. But I think about it fairly often, including literally earlier today. That's cool. I'd love to hear more of your thoughts as to whether or not there's this is a thing, right? I wonder if that's always been the case. It's just that we're more aware of it now. Surely there have always been regional stars, and these artists only stick out to you since they were in that sphere. That is true, but in terms of like who survives history, right? Because like the number of amazing scientists that exist in the world currently are numerous, too many to count. But the ones that the public will remember are the ones that get Nobel Prizes, and even then, it's only the ones that only people that read about Nobel Prizes will remember them as greats, right? But music, I feel like, in the age of radio, things were. I guess maybe that's the idea, is that the golden age of radio meant that everybody was tuning into the same few stations, so you could have a cultural cohesion. I see Penbu. So before the radio, where music was more regional, maybe it wasn't uh, quite that case. But I would also make the argument that, if we go back to like Baroque or whatever, classical music was defined by the tastemakers, people who had money, people who could commission these artists, and they were able to survive just on the prestige of their music, right? Sure, there were many great composers back in the day, but in terms of a canonical education for an intro to music class, you only hear about a fraction of them, right? And I'm wondering, of our millennial music, who gets to be Bach? I mean, there's Beyonce, Lady Gaga, T Swizzle. Yeah, yeah, I know. But I know that they're probably very good candidates if we had to put down an option. I just wonder how much of the population would agree, and is that percentage lower or higher than consensus on Elvis? Ironically, it looks to me more reminiscent of the musical landscape before the radio. Yeah, exactly. More micro fractured. I think that's always been the case. There was always a lot of pushback against, say, Elvis and the Beatles or what have you. That's true. Maybe we will more fondly remember these stars in a couple of decades, right? Because Elvis and the Beatles, these were countercultural performances that survived because they dragged the music scene with them, from what I understand, right? So maybe, even though it hurts maybe a little to believe maybe it is Taylor Swift, maybe in a couple decades that's okay to believe for the majority of millennials, but we'll just have to see. I do think fandom is more splintered than ever, but there is still a form of mainstream culture. Yeah, and I think it is easier to not participate in mainstream culture than it is ever before, because, I mean, hey, if you found me through the YouTube algorithm, that's YouTube being like, hey, you want to see a white blood cell explain biology to you, which is not something a lot of people want out of YouTube. So it is both like catering to our exact preferences these days, but also we're not all watching the same stuff anymore. Yeah, yeah. K-pop has taken a lot of inspiration from hip hop music as well. There's just a lot of music mixing globally. Yeah, and I think that's awesome that there's so much mixing. I'm just wondering, is there something qualitatively different about how much mixing there is, where you begin to lose the definitive edges around categories because we are innovating at speeds? There's no time to sit on new music, right? Like, how fast did mumble rap? Somebody who knows more about mumble rap confirm this for me. Like, was mumble rap like a big flare up and then just die immediately? Was there a lifetime? Is there still mumble rap today? Like, new people doing mumble rap? Or are we all like swirling different influences just in various combinations instead of experimenting with really, really unique sounds? I don't know the answer to that. Two hypotheses. Yeah, sorry. I want to acknowledge I am speaking from a very US centric perspective, right? You can apply this logic to your country as well. Like, I just naturally do it because I'm American and self-centered. Haha, American stereotype. America is the center of the world. But I think this argument is still very applicable to no matter what country you're from, right? You know, uh, not to say that the exact circumstances are the same. I'm just reporting what I'm experiencing as a music enjoyer who lives in America. Maybe you still do have musical generations in what country you're from. And if you do, like, tell me. I think that would be really cool to examine a living new genre, uh, uh, or one that is like mumble rap, where that's obviously a new genre, right? Mumble rap is pretty unique, I think. Yeah, see, I did say that comment because you did bring up an excellent point. I am being very US centric right now. Mariah Carey still singing in Christmas 2099. Oh, that's actually so good. You're so right. Mariah Carey, All I Want from Christmas Is You will outlive humanity. That might be. I think that's our Thriller, dude. I don't know if that's a millennial song, but it is like, yeah, that will survive until the end of time. Is the surprise voice of a generation, I hope. Surprise generation of biology enjoyers, hopefully, maybe. I assume the novelty of mumble rap has largely worn off, and there's still something not much. Yeah, that's an assumption, though. I don't really know that for a fact. I'm pretty sure klezmer music is still evolving. For one thing, you can find klezmer with non-Yiddish lyrics. That's interesting. I wonder what kind of music that is referring to. I don't know what that music is. U.S. music is still super influential here, so it does count. I do wonder how generations look like in South Korea, though. Yeah, like I wonder. Of course, I have my own hypotheses, right? That's why I find it interesting because I have a set of beliefs, but I'm very willing to like change my mind upon reviewing evidence. But I do think it's worth thinking about, right? Because there was a video, I didn't watch this video, but there was a thumbnail that was like, music is becoming more homogenous, that younger generations are looking towards older generations of music in search of novelty, that like because of commercialization of music, because producers are now chasing the chord progressions that give them the most money, there's like this optimal science of music that producers are trying to consciously take advantage of so that their music gets stuck in your head, because that's the ultimate goal, right? They want your money, and so they're going to make music that gets you to get addicted essentially to their music. I haven't watched the video. I don't know how grounded in evidence that claim is. I do kind of see how that could be true, right? There are certain progressions that produce different changes in your brain, evoke different sensations, et cetera, et cetera. So I do find it believable, but I don't know if there's hard evidence behind it. But this idea of like, because music from a capitalist perspective has been quote unquote solved potentially, like, the combination of chords does this thing in your brain that makes you give them money, that producers have been unwilling to try and find unique sounds that appeal, yes, to fewer people, but are more unique. And maybe, it's kind of like the whole perception of my channel, right? Where I specifically don't make my content accessible for everybody. I think it would be silly to claim that any YouTube channel is meant for everybody. My YouTube channel is for you guys, right? Nerds who like hearing about very high level things. And even though I could probably rope in bigger numbers by appealing to a wider audience, I find it better and more meaningful to try and provide a unique experience that fewer people will love. The problem is you get more money by appealing to more people. So I do understand this tension, and I do find it very interesting. That's not to say that good music still, doesn't exist. It's trends, right? Trends. I've been tempted by the music doomer mentality, but then An Evening with Silk Sonic by Bruno Mars and Anderson .Paak came out, and now IDK what to think. I'll have to give that a shot. I haven't heard that song. Sounds literally life-changing. And it's not to say all music will become bad, right? Because I think artists will always find a way to deliver on a song that you will love, right? Even if it is very corporatized. I'm not like that much of a doomer. I'm more looking at it towards like a cultural analysis perspective of like, are we seeing homogenization without a preconceived notion of whether or not that is good or not, right? I'm not making a value judgment. I'm just wondering what the data suggests, right? I heard there's a lot of big music performers whose music is all composed by singular songwriters who work in the background. I can imagine, like, people who are good at tricking you to spend money are good, right? Oh boy, AI designs psychoneural addiction composed music. Yeah, that's been a thing too, right? Like, a lot of companies are pumping out AI generated song tracks that look like they're working very well on the older generation, but honestly don't sound all that compelling to me. Because the thing about, and you know, this is maybe an individual consumer perspective, right? I'm not saying I'm better than any average consumer, right? But I think I like weirdo stuff. Like to me, the thing that like gets my brain all pumped up and excited is some really weird thing. It doesn't even have to be good at first, but just something to obsess and fixate over. Any deviation outside of normal to me is like nice, interesting. You know, that's why jokes work, right? That's why comedy works. Is, you know, there's a subversion of expectation and you laugh. And for music, I like it when there's like a thing to think about. And listening to some AI generated music, there's not like a thing, right? It sounds competent enough to my untrained eye. Like, I'm not claiming to be like, I can tell AI music versus non AI music. You know, I'm nowhere near that level. But what I am saying is that there's not like a thing that reads as coming from a weird person, because it is so perfect. It is so perfect without any imperfections. And imperfections are what give things their charming character, right? You don't want smooth edges all over, right? You need a little bit of roughness. And I think because, you know, when music is composed by a fewer number of people, you get more of that raw human roughness. You know, sometimes things don't sound all that good, or sometimes things are discordant, or, you know, or something doesn't sound the way that the chord is supposed to resolve in your head for the maximum amount of dopamine. And then, you know, conversely, because it doesn't resolve in the way that has the biggest neural payoff, that in itself becomes a novelty that you can get a different kind of spike for. And I think that's pretty interesting too. Yeah. Carol and Tuesday was documentary, changed my view. Yeah, interesting to revisit Carol and Tuesday in our current age. My hippie friends refer to it as the only good thing to come from recent pop. You're not a fan of APT by Bruno Mars and, oh, what's her name? Oh my gosh, what's her name? Is it, who did APT with Bruno Mars? Oh, K-pop stands gonna freaking roast me, dude. I love APT though. And the thing is I think APT is a lot of charm, right? It's an earworm, and but I think APT for me stands the test of time. I think I don't get tired of that. I think I can play it at any time and get happy. I'm not a doomer, but I also accept that sometimes I just need to look a little bit harder. Sometimes it's best to not look a bit harder and just have your mental health be kept intact, because like, I'm not associating a value judgment with this philosophy, so I'm not negatively impacted by it, because I do have the belief that artists are going to art. You might have to work a little harder. They might not get paid as much. There might be less of it. But I think a part of the indomitable human spirit is that there's always going to be somebody putting art out there, and that art is going to be good, because our brains have evolved to the point where we have to find meaning, or at least, you know, we don't have to find meaning, but it is so smart that we can prioritize finding meaning outside of just putting food in our mouths. So, you know, I'm not that much of a doomer, I would say. Thank you for subscribing, Mr. Sandwich. I want novel experiences. Yeah, like, and the indomitable spirit is novel experiences, right? There's a, I think, I don't remember who did it, either CPG Grey or SciShow, about the spirit of human exploration. That novelty, that ability to explore, is what broke us out of the Fertile Crescent, or whatever, wherever humanity emerged, right? The ability to cross oceans, right? Novelty. Humans are great at that, and I don't think that'll ever stop. We're always going to seek novelty. AI garbage music is only going to get more homogenous and terrible. And newness is always going to be attractive, right? I'm not saying AI music will be AI music will get more indistinguishable from human music, but AI can only copy, right? It can only facilitate a facsimile of a human experience. And I think something to be happy about is that because humans are imperfect, like AI, okay, this is what I mean to say. AI, the goal is perfection. It is to assimilate all of the human experience into one consumable perfection. And because perfection cannot, by definition, perfection cannot have rough edges, you are still going to get humans making rough edge art. And that rough edge art, I think, will always be valuable. I hope. Right? Some boomers on Facebook might make me doom when they go, oh, this music sounds better than 90% of the music written today. Those boomers, okay, I feel sad for them. I hope that in the future, not counting those boomers on Facebook, that novelty will be prioritized. Yeah. My hot take is mid-music has always existed. AI just made it 1,000 times more common. Yeah, for sure, right? That's why I'm talking about greats. Like, there's a lot of music that you like, some music that you love, but I think most music you don't really care about. And, it's hard to make music that people like, right? It's, as someone who is playing with composition, it is hard to get something that not only sounds good, but also that people will like. Because, a nuance that I think I came to, conclude with Penbu is that you can string together a nice sounding chord progression. And as Penbu showed me, it's actually not that hard to assemble a progression of chords that to the ear sounds pleasing. I think the difficulty of making good music is having that be the basis of something of art that people will love, right? And the thing about imperfection is I think there's a part of our psyche that loves imperfection because it is a reflection of a connection to another human, right? Because we are imperfect. We see imperfection in other people. We are drawn to share that level of imperfection. At least that is my hypothesis. I don't claim to know that for a fact. But because AI will be perfect, you can't relate to perfection, dude. You can't relate to perfection. That's not relatable. And of course, content has to be relatable. Looks like it was Rose. Aha, thank you, Penbu. It was Rose. Yeah. Even if AI music is itself just as good, I think knowing it wasn't made by a human makes it a different experience, at least to some people. This is the argument that I made in my members only video about AI art, where AI bros want to force you into a false dichotomy of believing that that art should be separate from the artist. They want you to buy that premise because, the thing that matters is the pixels on the screen and the reaction that happens in your brain when you see those pixels. That's the only thing that matters about art. Disconnect yourself from the artist. And I think that is not a good way to live life because that is a very lonely way to view art. Art, to me, is the expression of a human being. And as a person who enjoys art, you are sharing a very intimate moment with a very intimate portion of a person's brain. Right? They are opening up. They are showing you my inside thoughts. Or they are showing you what I believe to be good art. Right? If I put a 5 video out, it comes with an endorsement that I think that is good art. And so by you watching the video, you are seeing my personal standards. You are seeing my personal artistry. And I think that matters. I think it matters to me that you know that it came from me directly, which is why the team is so small. It's just me and Toxo. And Mylan sometimes contributes art, which is great. But for the most part, it's just me. And if you were to remove that kind of element, or, artificially in your head, disconnect the art from the artist, you are losing the ability to feel connected with the artist themselves. And I think that's important because we are in an age where modern connection online, the globalness of culture these days, again, severs you from a larger group. But if you all look at the same image and you all try to see what the artist was trying to get through, you actually put yourself in their shoes, which is an act of empathy, right? When you look at a Picasso painting, it's not just, looks cool, I guess. I mean, you could enjoy art that way. There's nothing wrong with that. But I think you get more out of it by going, what was Picasso feeling when he drew this painting? What does this painting mean to Picasso? if I were Picasso and this came out of my brain, what would that mean for my emotions, right? It's a practice in empathy. And this extends to all forms of art, I think, right? Not just paintings, right? A piece of music, right? A piece of music that somebody composes is their way of showing you, this is what I think sounds good. This is the song that I wanted to write. And you might think it's cringe, but that's okay because art is cringe, right? Good art is cringe. Good art is cringe because it is personal, and it is cringe because some number of people are not going to find the inside of your brain all that appealing, which is fine. But some people are going to deeply relate to it, and they're going to love it, and they're going to feel connected to you, and you're going to feel connected to them. And I think that two-way connection is very, very important. Very, very important. Yeah. So I agree with you, James, right? Like, I really dislike the attitude of, oh, just, art and it doesn't really matter, separate the art from the artist. And I think you just lose, like, you lose an aspect of the human experience, right? You lose an ability, a venue by which you can connect to a human being. And I love art because I can feel connected to a human being without having to talk to them, because I am asocial and very introverted. and strangers make me nervous, right? Having a two-way exchange with a stranger where I'm constantly in entertainer mode, where I'm always worried if I'm being entertaining or not, or if they're just zoning out, because you can imagine I'm not like a hit at parties. I don't even know what people talk about. Okay, so I do know because I've been paying attention, right? I study these conversations because I feel so out of place at parties sometimes. But when I'm at a party with people I don't know, I take note of what they're talking about. Oh, it's like stories. So they have interesting lives. They're doing interesting things. I don't live like a very interesting life. Apart from YouTube now, I like attempting to make YouTube work. I don't think I have all that much going on in my life where, I don't have a trip to Barcelona, right? I don't have this abroad experience. I don't have a funny story about a drunk night at a bar, right? I don't have these things, these experiences. What I do have is the ability to hyperfixate on one idea and then yap about that for, however long. And for some people that works very well. For others, the most boring thing in the entire world is talking about whether AI art is making all art more homogenous, or how algorithms prune or hyper optimize dopamine rushes. A lot of people are going to find all that crap really, really boring. But what I mean to say is that, like, because there's more than one way to feel connected to another human being, art is such an important part of that ability, right? You don't literally have to be in the same room with somebody to feel connected with them. And being like, oh, separate the art from the artist, you are preventing a genuine human experience, and that sucks. Certain people have the ability to describe relatively mundane experiences in a way that's interesting. I think it's more often about the delivery versus the content of the story. Some people are very gifted at they have a rhythm of speech that is addicting to listen to. I do agree with that. But that also comes from like the buildup of confidence of like, having the confidence that your delivery style does work for a lot of people is good. Whereas for me, I do better when I can find a weird thing that you also fixate on, and then we can fixate on together. Or you can teach me why you fixate on a certain thing, right? I am terrible in situations where it's like, you go home, and if you're not obsessing over some kind of idea, you just live your life, I'm gonna have a very hard time trying to not bore you. And I've gotten advice before, you do more listening, but then I don't know what to say in response, because if someone is telling a funny story, I often find myself just going like, man, that's crazy, that's so funny. I just don't have anything to add. I'm not quick or clever enough to add on to stories. Follow-up questions, I think that was a good piece of advice. I do try to do that, but I feel like sometimes I can get too in the weeds with follow-up questions, and then I can distract. If they have to take a break from telling their story, I can ruin their rhythm. You can feel all this kind of feedback in the way that people emote. Yeah, so that's also why I enjoy talking to like academics, because even if they're not in the mood to talk about their research, they're usually obsessing over something, because I think you have to have some level of obsession going to graduate school. It could be cultivation patterns in farms in Inner Mongolia or whatever. But if you get a chance to talk to an academic, a working academic, they could just yap. And I think to me, it's much easier to find interesting questions to ask about whatever they're obsessed with versus the actual details of their life, which I'm really bad at. Are you familiar with the solid snake method of conversation? I am not. What is that? Care to enlighten me? That being said, like now that I am a YouTuber, the number of new people I talk to is like zero outside of like finding new collab partners. Like the number of parties that I go to, sharp decline, sharp decline after leaving my real job. It's when you just repeat what you heard, but as a question. I don't really know. I don't really know if I like that. I don't really know if I like maybe to buy time to come up with something interesting to say. As a stall method, that might be kind of fun. Yeah. You don't know that if, oh god, it's happening. Every kindergarten class is converging. What just kindergarten children repeating each other ad nauseum. Okay, yeah, I wanted to leave you all with that idea, the idea of this like cultural homogenization and the death of distinct new genres of music and homogenization and all that kind of stuff. Because I come back to this analogy a lot of the big rip idea of like the universe is expanding. Is it exponentially expanding? If it's exponentially expanding, then light from the origin of the universe will never reach the edges. So there's this like black hole that you'll never be able to escape. Not literal black hole, but like a conceptual black hole that you'll never be able to escape. And I'm wondering if we're in the big rip era of culture where new culture is being made so fast that there's no opportunity for it to become big anymore, just big in a micro community. And again, no judgment as to whether or not that's a good or bad thing, but I wonder if it's happening. You're gonna sound like Meowth when you translate what another Pokemon is saying. Yeah. That's a pretty funny idea. God, the choice to give Meowth the accent that Meowth has was certainly a choice in the English dub, or at least in the US dub. All right, that's gonna be it for me. I feel like the pent up yapping, I have finally been relieved of the desire to do this pent up yapping. So thank you all for listening to me. Thank you all for attending the stream on the very important contributions that the cat has had on the understanding of our visual system. Again, our two shout outs today are that I'm going to have a 12-hour charity stream in January, probably January 10th. And then one more plug for a collab that I will hopefully be able to be an audience member of with Z Insider that is occurring tomorrow. And with that being said, I hope you all have a good rest of your days. And I hope to release the giant virus video relatively soon, hopefully sometime next week. All right, that's it. Bye, y'all. Bye-bye. Bye.